to do this activity. As you can see, the nest building score, we have a way to score the kind of nest that they make. The animals which did not get the drug had a much lower nest score than the animals which were given the drug. And our drug 2024 was given for 9 months. So it's a very slow improvement, but we are actually seeing a slow and steady improvement in these mice. So now I'll quickly show, this was data with 2015. Similarly, this was the ability to find a platform. It was again another memory task. As you can see, the 2015 molecule improved the ability to reach the platform that we need to reach much faster than the mice which did not get the drug. Similarly, the nest building score was much better with animals which got the 2015 diet. We gave it in the form of a diet. So overall, the conclusions are that we have two fantastic compounds, 2024 and 2015, which are centrally active. They are orally bioavailable and centrally active and engage our desired target, which is brain TNF-alpha. We are lowering brain TNF-alpha and improving cognitive function. So, and as I'll reiterate this point one more time, that our oral PD2024 and 2015 molecules decreased tangles in independently published studies. So this was done separately. One was done by us in collaboration with UCLA, and one was done at the NIH. And both these studies showed the same thing with these molecules. Our compounds significantly improved cognitive and behavioral symptoms. And finally, our compound decreased phospho-tau pathology and brain TNF-alpha levels in a FTD model as well as an AD model. So those are the conclusions. I wish all the students the very best. I think I've run over my time, Professor Ramesh, or do I have a few more minutes?
No, sir. No problem, sir. Thank you, sir.
Thank you. I'd be happy to take any questions. I hope this was a talk that many of you were able to relate to, because I know it's not classical zoology, but it's hopefully something that was interesting enough for you to become interested in drug development.
Yes, sir.
Thank you.
Spurti, can you? Spurti?
Yes, sir.
Was interesting enough for you to become interested in drug development.
Yes, sir. Please put down your questions in your chat box. We will read the questions and then let it down to the speaker.
Sorry, I have already put one question. If you allow me, can I ask a question to the speaker?
Yeah, okay.
Good morning, sir. This is Jayashri Vadia. I just want to know whether the same drug, similar to the same drug, can be used for Parkinson's.
Good question. Very, very good question. Actually, we have tried it in Parkinson's disease. So Parkinson's, so the whole point, Professor Jayashri, is that your question?
Sir, is only DBS.
Whose question was this?
Voice is breaking, madam.
Sir?
Yes.
Is DBS is only the treatment or any other treatment for Parkinson's?
You mean deep brain stimulation?
Yes, sir.
Yeah, so I would say for Parkinson's, there are not any disease modifying treatments. So there is nothing that is reversing the disease yet. Right now, they're only treating symptomatically. Now, there are some genetically based forms of Parkinson's, like the synucleinopathies, where there might be more progress that has been made. But overall, I would say that there are no disease modifying treatments for Parkinson's. To your point.
Thank you, sir.
One quick point I'll make is We have actually tried our compounds in Parkinson's disease, and we have seen some target engagement, but not clear-cut data to show that we've improved motor symptoms. We have not seen that yet.
But Alzheimer and dementia, they are improving with this drug.
Yes, yes, very,
Oh.
very clearly, very repeatably. We have done some of these experiments twice or thrice already, and each time it has done it well. It has improved the cognitive symptoms.
Sir,
Oh,
last question. Is it due to polygenic?
I'm not sure what you mean by that.
Parkinson's is a polygene inheritance. So is it this type
So
of drug?
Parkinson's, most for Parkinson's, I can say that maybe less than 5% is genetically based. Most of it is idiopathic. They are not clear on the exact reason for why a person gets. It's not just genetic. Very few, the much smaller percentage is genetic. Now, coming to what I talked about today, the frontotemporal dementia is a very genetic based disease. So that's actually easier to develop a treatment for. Easier meaning it's a much more clear cut population that we can recruit to treat.
Okay, like microglia activation can also be done in Parkinson's.
That happens. Yes, it happens. But is it the question is whether it's an epiphenomenon or is it an underlying pathology? That's the issue, right? I mean, is it happening around the disease? It may have nothing to do with the disease. It is something that happens, kind of a peripheral thing that is happening. It is not driving the disease. The question is, when you're developing a drug, you want to block something that drives the disease, not something that happens on the side.
Surrounding the substantia nigra, the microglia surrounding the substantia nigra, sir?
Yes. So, I mean, very fair point. But the question is whether we have, in a model, we have to see whether that is actually going to block the motor symptoms.
Thank you so much, sir.
Thank you, sir.
Thank you so much, sir.
Murthy, any other questions are there in the chat? Yeah. Madam, please mute your. Spurti?
Yes, sir. I'll be reading out the questions from the chat box. Hello, sir. Are you able to hear me?
Yeah.
Yes, I can hear you fine.
Yes, sir. There is one more question. Sir, are you able to hear me?
I can hear you just fine. Go ahead.
Read the question.
Yes, sir. Is the drug has an impact on some other glial cells? The question.
We have not looked at that. We've not looked at, so basically astroglia, that's the only other cells that we can look at. We have mainly looked at microglia. Astrocytes, yeah, it's likely because those are also producing inflammatory cytokines. So we would expect the overall decrease in inflammatory cytokines.
Yeah.
Next question.
Next question, Spurti, go ahead.
With such wonderful results, when is this drug possible to roll out for clinical trials?
As soon as I get money to take it to IND.
Yeah.
You need to tell people to mute their.
Yes, sir. Is Alzheimer's disease a genetic disorder or is it due to mutations?
Joe, I mean, so there's a much smaller percentage of genetic mutation derived disease, but I hope I can. You can hear me.
Oh. Spurti, ask them to mute them. Spurti, mute them. Yeah.
Good. Thank you so much, Spurti.
Thank you, sir. Thank you for your
yeah, did I answer your last question? Yes, but I'll make it quick. The
Yes,
number,
sir. How long will
the
it take to
majority.
see the benefits?
Go ahead. Go ahead. Please continue, Spurti.
Continue, Spurti.
Did I answer your last question? Yes, but go.
Rebound.
I'll make it quick. The number, the majority. Go ahead.
Spurti, go ahead.
Please continue,
Spurti.
Sir, I guess there is a lag between the live YouTube session and this, sir. I'll just get back in 1 minute.
No problem. I'll wait. I can wait.
Okay. Thank you, sir.
Can I proceed, sir? Ramesh, sir?
No, Spurti is having one question. Yes, Spurti?
Yes, sir. There's this last question.
The last
You are
question.
using the compounds for a long term in mice. If we can extrapolate that time period, it will be a long term treatment in humans. So will it be considered as a preventive drug as a treatment?
Great question. Great question. So ideally, you want to give a drug only when there are symptoms start showing up. But now more and more, the FDA is looking for drugs which can prevent the disease from coming, from actually emerging. So the symptoms from emerging. So we'll go where the data leads us. If we are able to, we'll be lucky enough to take it to the clinic. We'll go where the data leads us. If we want to start earlier treatment, because there is a phase before Alzheimer's disease called mild cognitive impairment. So if people can take the drug when they are diagnosed with mild cognitive impairment, then they may not get full-blown Alzheimer's disease. So there is a phase before Alzheimer's disease, mild cognitive impairment.
Yes, sir. Now I would like to request Shakuntala ma'am to deliver the vote of thanks.
Yeah, okay, Spurti. It's my privilege to thank Dr. S. Prasad Gavita, CEO, Research and Development, P2D Biosciences, Ohio, USA, for his keynote presentation on drug delivery and development by treating neurodegenerative disorders. You are given a valuable information on dementia, rheumatic arthritis, colitis, and Parkinson's disease, and their inflammatory action, and also the drugs to cure these diseases. Sir, the effective and advanced drug development for neurological disorders is the need of the moment, like to cure mucormycosis, a dreadful disease affecting the people in India after COVID-19 infections. We are fortunate enough, you sir, in this webinar session, to have you in this webinar session in terms of acquiring knowledge in this field. Thank you for enriching us by your informative presentation. On behalf of our management and organizing committee, profusely, I thank you, sir. Thank you once again.
Thank you.
Hello?
Thank you, sir. Thank you.
Thank you, Spurti. Thank you, Professor Ramesh.
Thank you so much, sir, for sharing your knowledge.
Thank you, sir. Yes, Spurti. Yeah.
Thank you, sir. Now I would like to request Professor Ramesh sir to welcome Dinesh sir.
Good morning, sir. Good morning to all of you. It is my privilege to welcome the most dignified resource person for today's national international webinar, Professor Dinesh Kumar, Professor and Chair, Department of Plant Biology, College of Biological Science, Genome Center, University of California, USA. He obtained his PhD in molecular cellular developmental biology from Iowa University in the year 1994. His ongoing research includes post microbe interactions, NLRs, and PRRs functions in innate immune signaling, interorganal communications, programmed cell death, autophagy, virus host interactions, gene editing, grade group affiliation, biochemistry, molecular, cellular, and developmental and plant biology. He specialized in cell and developmental biology, molecular biology, biochemistry, and genomic, environment, and integrative biology. He has received several honors and awards. Some of them includes uh, Neil T. Keane Award for Research Excellence in Molecular Plant Pathology, American Phytopathological Society in 2019. Association for Advancement of Science Fellow 2018. Faculty Research Award, College of Biological Science, UC Davis 2015-16. Semi-finalist HHMI GBMF Investigator Competition 2010 and 11. Faculty Fellowship 2002 to 2003. Hellman Family Fellowship 2000. Life Science Research Foundation Postdoctoral Fellowship 1994 to 1998. Sir is also having, already our President Dr. H. Ramarao mentioned that he is associated with American Society of Plant Biology and American Phytopathological Society. He is having a credit of more than 25 research publications at national and international journals and members of different other societies. It is my privilege for all of us to have such a great, wonderful, versatile personality among us. We all are eager to know about your research work, knowledge, and path. Thank you for sparing out time for all of us from your busy schedule to share your knowledge with. Once again, welcome you to this international webinar, Dr. Dinesh Kumar, sir. Now he will continue on autophagy and beyond. Hearty welcome to the webinar, sir. Thank you for our invitation and accepting.
You hear me?
Yes.
Yeah, let me. Can you give me permission?
Yes, sir. Just a second.
Yeah.
Yeah.
Yes, sir. You're now the co-host. I would like to request everyone to please mute your microphones because it is causing a lot of disturbance to the speaker.
Yeah.
Thank you once again.
Yeah.
Can you see my slides?
Yes, sir. It is visible.
Okay. Yeah. So I like to first thank Professor Ramesh for the great introduction.
It is my privilege, sir.
And also for invitation to speak here, and also Professor Dr. Ramarao for the initial introduction, actually. So I may be a little bit of out of topic here, but hopefully by the time I finish, you realize that plants, humans, and insects, they all do similar thing, but using a different platform, actually. So my long-term interest in our group is how do actually plants able to defend against pathogens similar to how humans do, actually. But remember that the plants cannot move. So they have to do some more things. Unlike us, we can actually move. For example, during COVID-19, some people fled where there was a higher incidence, but plants cannot do it, actually. But the other thing you need to remember about plants is that the plants don't have immune cells, unlike us, where we have different immune cells to take care of the infection, actually. But every plant cell has the ability to defend against the invader, actually. So that's the distinction between plants and animals and humans, but I will show you that they all use similar molecules actually. So just to give you some perspectives on why study plant diseases is that, let me see if I move this. Okay. So 15% of the global food production is lost due to diseases every year actually. And this is very common in India also. The classic example one uses is this Irish potato famine caused on potato by the oomycete pathogen. Actually, about 50 million people migrated from Ireland to United States because of not having food and caused by this specific disease in 1800s actually. But then in the recent years, this is an issue even in India, is this Uganda strain of the fungus, which is causing a major problem in wheat actually. Maybe a little bit of a problem, but it is a major problem in the United States right now. Is this what is called a citrus greening disease? Expectation is that if we don't come up with a solution to this disease, we will not be able to eat oranges and drink orange juice in the next 20 years or so actually. And then finally, India and also mainly in Sub-Saharan Africa, some of the people in Sub-Saharan Africa entirely depend on this crop, cassava actually. So if they don't grow cassava and make some money, not only they cannot eat, but also they cannot send people to schools actually. So these viruses causes major disease in cassava, causing about $2 million of loss annually just because of these diseases here actually. So this is a platform why, just to give you an example of why one should study diseases in plants, similar to what we do in humans. But my lab primarily works on very basic research on understanding what causes diseases and how actually plants able to defend against these infections actually. So if you look out your windows, you see that a lot of plants are healthy and they are perfectly fine actually. So the immune response by plants is enormous actually. Every plant can actually defend against the infection. So that's the reason why you see in the nature, plants are completely fine actually. So that the disease is not an arm in plants actually. So only if there is a perfect environmental condition and if a pathogen has highly evolved, then it is able to cause disease actually. So just like in humans, like in us, plants also have these what is called as cell surface receptors actually. So the function of these receptors in the cell surface is to recognize the general features of the pathogen. So for example, say bacteria has flagellin. So there is a dedicated receptor which recognizes this flagellin, not only in plants, but also in humans actually. And in plants, when these receptors recognizes these features, activate an immune response by signaling and turns on gene expression, and that is what gives you the resistance actually. Similar to again, what happens in humans. But of course, you do see disease in the field actually. So that is because highly evolved bacteria, for example, is able to use this injection system to send proteins of its own into the host cell actually. So these are often referred to as effectors. So the main function of these effectors is to interfere with this immune pathway actually, so that it can shut off immunity so that the pathogen can survive actually. But of course, the plants are smart. In evolution, they have evolved with a second line of defense, so which are intracellular receptors. So these receptors are often referred to as NLRs, and they recognize these effectors and activate a robust immune response to contain the pathogen to an infection site actually. So basically, the plants have these two layers of defense. One is a cell surface receptor mediated, and another one is an intracellular NLR mediated defense to pretty much able to defend against any pathogens actually, okay? So my lab is primarily interested in this area here, but before I tell you what we do, interestingly, when we discovered these receptors, it became apparent that these receptors are common between plants and other organisms, actually. So as I just alluded, the cell surface receptors recognize pathogens to activate immunity in plants. And similarly, in Drosophila, there are cell surface receptor with similar features, except there is a difference in the N-terminal intracellular domain called toll-like receptors to activate immunity in fruit flies, for example. And actually, once we discovered these molecules, which are similar to Drosophila in 2000, about a year later, the human people discovered that the toll receptors are also there in the humans, and their function seems to also recognize the pathogens to activate immune response, actually. So as you can see here, although the plants and humans and fruit flies have diverged quite a long time ago, but they are all using similar kind of molecules to activate an immune response, actually. So basically, this immune system is pretty much conserved across different organisms here, actually. And then, of course, as I said, the plants have these intracellular receptors. But in fact, the mammalian system, humans also have about 50 or so intracellular receptors. These are highly similar to the receptors in the plant, actually. So as Professor Gavita indicated that the Crohn's disease in humans is caused by mutation in one of these receptors called NOD receptors, actually. So these NOD receptors are highly similar to the plant intracellular receptors. In fact, functionally, they behave similarly, actually. So you can appreciate that the immune system, although we work on plants, the knowledge we gain from plants is translatable. Plus, we use quite a bit of knowledge gained from the mammalian and fruit fly to infer ourselves in the case of plants. So the primary goal in our lab is to understand how the so-called and the intracellular receptors actually activate immune response by what kind of signaling. So pretty much my lab is primarily interested in basic research in understanding these responses here. And you can refer to some of these papers which are shown here if you want to learn more about it. But I will just give you highlights of what kind of work we do, actually. So recently, so we've been working on this project quite a while, and others are also working on this project quite a while. But we have not been able to identify many players in this signaling platform because of the technical difficulties. I'm not going to go into the detail. In order to overcome that, recently, about a couple of years ago now, we discovered this proximity labeling approach to identify proteins that these receptors interacts in order to defend against this specific virus here, actually. So I'm not going to go into the detail, but the idea is that this approach allows you to identify a number of proteins that potentially play a role in this signaling platform here, actually. And we focused on one of the protein called UBR7 and showed the function for UBR7 in this pathway here, actually. So the reason we focused on UBR7 is that it is an unknown protein which is involved in degradation, protein degradation, actually. So I'm not going to go into the details on the details of this specific study, but you can look into this here in this paper here. But 1 year after we published this study, it became apparent that the human pathogens also use an UBR like E3 ligase in order to defend against the pathogen, actually. So what you can see here is that the approaches we take to identify partners seem to be similar to what are involved in the human innate immune system also. So what we speculate based on what we discovered about this UBR7 is that, so you don't want to have a receptor, immune receptor, which is activated, actually, or you don't want to have a receptor which is active, actually. So the reason for this is that this is the main reason why you have autoimmune diseases in humans, actually. So if you were to activate this in the absence of the pathogen, the plant is going to kill itself, actually. So of course, plant doesn't want to do it. So what it does is that it borrows this protein E3 ligase to degrade itself, actually. So when the virus comes in, that virus somehow interacts with this ligase here and disrupt this interaction so that this receptor can now become stable and then activate an immune response, actually. So this is one way of avoiding autoimmunity or killing itself to survive, but defend when the pathogen actually gets into the plant, actually, okay? So I'm going to switch gear here from studying this signaling platform and protein interaction to the interorganellar communication during immunity, actually. So a few years ago now, we discovered that there is an interaction between the chloroplast, which is a dedicated organelle in plants for photosynthesis. So that's the reason why plants are green. And that's the reason why we can get food from the plant, seem to directly interact with the nucleus in order to activate immune response, actually. So this is very new, and this interaction was not discovered before. And similar kind of interaction in human innate immunity between the nucleus and the mitochondria, actually. So although it occurs in plants, but main interactions seem to be between chloroplast and nucleus, actually. So while studying these processes, we bumped into the observation that when the plant is activating an immunity against the pathogens, it actually changes the chloroplast shape. So if there is no pathogen, so the chloroplasts stay round, you can see here. But when the immune response is activated, you see these structures that are coming out of the chloroplast, and these are referred to as stromeals, actually, okay? So not only the structures are coming out during immune response, but they're also making interaction with the nucleus, actually. So what you see in the yellow here, blob, is a nucleus of the plant cell. And you can see these structures, stromeals, wrap around or making contact into the nucleus during an immunity, actually. But this is a highly dynamic interaction. As you can see, a chloroplast is sending out a long tubular structure into the nucleus periphery to make an interaction, actually. So these observations were not made until about 3 or 5 years ago, until we made it. And then we showed some of the phenomenon for the function of these, actually. So in the last few years, we've been interested in understanding this, the finer details of how and why this kind of interaction occurs during plant innate immune response, actually. So as you can see, this is the dynamic nature of these stromeals. Need to have some kind of a structure from the plant which can support this dynamicity, actually. So in fact, what support this dynamicity is the cytoskeleton. So probably you have learned in your classes that the cytoskeletons are like actin and microtubule play an important role in cellular function, actually. So the chloroplast stromeals seem to be formed and being associated with the microtubule, which is shown in yellow here, is what keeps them that dynamic, actually. If I play the movie here, you can see they don't leave the microtubule attachment at all, actually. So wherever they're going, they're always in support with the microtubule here, actually. So what we have shown in this eLife paper we published a couple of years ago is that not only the microtubules are important for the formation of these structures, but they're also important for guiding the chloroplast towards nucleus, actually. And in addition to the microtubule, the ER structure in the background, actually, of the cell supports these structures, actually. So what you see here is in the magenta is ER, in the yellow is microtubule, and then the chloroplast is blue, actually. So if I play this movie, as you can see, the dynamic nature of the ER here, but the chloroplast stromal is following the microtubule, not the ER structure. So if the microtubule comes out of the ER structure, as also the stromal comes out of the ER structure, actually. So what we think is happening here, based on all the observation we have made, I'm skipping a lot of data here, is that these cytoskeleton interaction with the chloroplast is important to maintain this interaction with the nucleus, actually. So what you see here is the center is the nucleus, in the background is the microtubule. So the chloroplast is attaching to the nucleus and maintaining this dynamic nature during an immunity. And this is the reason why you need these players here, actually. But of course, the big question is why? Why do you have to do all these gymnastics in order to activate an immunity, actually? So the answer to that is we don't know, actually. So we are doing a lot of work on trying to understand why this interaction need to be maintained, and why this dynamicity need to be maintained, what are the players, what genetics is involved here, and what genes are involved here. Of course, finally, why do this actually? Okay. So if you're interested in this topic, you can look at these two published papers. I'm not going to go explain this, but we were fortunate to have a cover page article when we first discovered this phenomenon actually. So I'm going to switch a little bit here and tell you a little bit of application. Although my lab is heavily into the basic cell biology and basic signal transduction, occasionally we bump into an idea that leads into some of the application actually, because if you see our work, we always on the side develop a modern technologies that we can use to answer the questions in immunity actually. So one question which we are interested in the lab and also others in this area is that when the immune receptor recognizes the pathogen, activate a defense response through activating genes, which ultimately leads to killing itself actually. Okay. So when the pathogen, for example, if you put a pathogen here, number of cells where the pathogen has infected is going to kill itself actually. So this is the way of self-sacrificing to make sure that the rest of the plant is free from the pathogen actually. So it kills itself because it deprives the nutrient for the pathogen. So pathogen cannot go out of this dead cells. So it's restricted there. So the rest of the plant can be happily lived after that actually. So, but the question always is that, you know, there are about 30 to 50 cells die actually. After that, it doesn't move. So initially pathogen beginning to establish it, but plant responds it. So it kills itself. So the pathogen is contained. But our question always has been, why not kill some more cells? Why only these number of cells actually? So in the process of identifying these players, by doing reverse genetics, we identified a major role for autophagy in this process actually. So as you can see here, if we label the virus with a GFP, wherever the virus is there, we can monitor it, and we can also monitor the death actually. So basically wherever the virus is, you see the death. So virus basically does not move out of here. After 2-3 weeks, the virus gets cleared actually. But if you knock out a gene which is required for autophagy, then the death seems to keep spreading actually. So death is not contained anymore. So what we discovered is that in fact, this is the first discovery in plants before it was discovered in the mammalian system. So autophagy seemed to be required to limit the death to the infection site during immunity actually. So what is autophagy? So autophagy is basically what happens between your meals actually, okay? So this was first discovered in yeast as a response to starvation actually. So if you starve the yeast with, don't give the nitrogen actually. So what yeast does is yeast can actually survive for a quite long time. That is because it activates this autophagy process. And what yeast does is that it actually takes part of its own cell, encloses this in autophagism structures, digests it, and give it back the amino acids to the yeast to survive actually. And in fact, nobody believed this almost for 20 years about the work from Osumi in Japan actually, until people started to find the similar phenomenon in humans and in plants actually. And in fact, for that, almost 25 years later of discovery of this process, Yoshinori Osumi from Japan was given Nobel Prize actually. So in fact, if in between your meals from lunch to dinner, you're actually activating this process to digest the material actually. And this is the reason why the longevity these days, people have been studying that, you know, if you starve occasionally, your lifespan is going to increase actually. So when you are starving, it's actually going to activate an autophagy and takes part of your cellular materials, digests it, and giving back to yourself actually. And in fact, it has much more role than that. I'm going to come back to that soon in probably the next slide. Yeah. So just to introduce here these topics, because it's important for what I'm going to say in the next few minutes. So I'm not going, so there are about 30 genes are involved in this process. It's not important to remember any of those actually, except for two. So initially, there is an initiation process. This initiation process results in expansion of the autophagosome actually. So this initiation and expansion requires two key proteins that is important for us to remember for today's discussion. One is ATG8. It is an ubiquitin-like protein which has a glycine at the C terminus, which is what actually determines the phagophore formation and also determines what needs to be enclosed in this autophagosome actually. But this ATG8 first has to be processed. So in order to process this, you need a protease. This protease is ATG4. The ATG4 basically cleaves after the glycine. Once it cleaves, ATG8 is ready to make these structures and recruit the cargos. Actually, of course, this is a very simplified version. And then it targets this phagophore once it is formed with the cargo into the vacuole in the case of plant and yeast, or lysosome in the case of the human cells. And then the cargo is delivered into the vacuole or lysosome, and the enzymes in these organelle is going to digest, either getting rid of them or it gives back the material back to cell actually. So one example of the use of this process is that, for example, say you have a damaged mitochondria actually. So you don't want to keep the damaged mitochondria because then you are going to activate ROS, which is going to start an inflammation actually. So the reason you get rid of mitochondria, which is damaged, is because of the autophagy actually. So mitochondria is recognized by ATG8, encloses this in autophagosome, targets to the lysosome, and degrades it actually, so that you can reduce the inflammation process actually. So now, in the last 10 years or so, it's very clear that the autophagy plays an important role in not just starvation, but also in many, many different aspects of biology actually. So Alzheimer's that you just heard from Dr. Gavita, Huntington diseases, innate and adaptive immunity, innate immunity in plants, nutrient recycling, aging, senescence, cancer. You can name anything that the process seems to be involved actually. But the interesting thing I want to talk about today is that autophagy is also important for plant pathogenic and the human pathogenic fungi to cause disease actually. So one pathogen that we have been studying is this pathogen fungus botrytis actually. So you're probably all familiar with fungal mold in your home actually. So if you were to leave a strawberry or tomato on the kitchen table, 2 days later, if you come back, you see this mold developing on those fruits actually. So that is mainly because of this fungus called botrytis actually. So interestingly enough, for botrytis to form the disease, it actually requires autophagy actually. So if you knock out, so this is the work done by Jung Chen Wu in the lab, a former member of the lab, is that if you knock out autophagy, the botrytis does not cause disease anymore on the tomato leaves as opposed to the control here actually, okay? So this gave us an idea that maybe we can target autophagy as a tool to control these diseases, potentially developing a fungicide actually. So the reason for that is this. Remember I informed you that a ATG4 protease cleaves ATG8 in order to activate ATG8 actually. So the idea here is that if we can able to interfere with this cleavage of ATG4 by ATG8 by ATG4 in the botrytis, then we can actually able to stop the botrytis growth actually. So that was an idea. So we purified these sensors. I'm not going to go into the details. So the idea of this sensor is that ATG8 is fused to a fluorescent protein called citrine and a luciferase actually. So if there is no cleavage, you get very high activity of citrine actually. But if the ATG4 cleaves ATG8, then it becomes two parts. So you lose the citrine fluorescence actually, and you also lose the luciferase activity. So this gives us a very nice screening platform in vitro actually, because we can purify ATG4, we can purify ATG8, mix it and add a drug and identify a drug which can interfere with this cleavage here actually. So this is basically what we did. So we add the small molecule drug, and then we add the ATG4, we wait for 10 minutes, and then we add the ATG8 substrate, and then we add the substrate which can shine the luciferase, then we measure in this spectrometer actually. So by doing this screen, about 3,000 chemicals, these are coming from NIH library, we were able to identify chemicals which interfere with cleavage of ATG8 by ATG4 actually. So I'll just show you one example here. The chemical name I'm calling it H4. So if you don't have H4 in the control, you can see the ATG8 is completely cleaved by ATG4. But if you have the H4 chemical, then the cleavage is completely interfered. Okay. And then if you use the same chemical here at different concentration shown here, in the mark, the botrytis spore can germinate in the plate, but in the presence of this chemical, botrytis spores cannot germinate actually. But of course, we have a lot of other in vitro data. I'm not going to go into the detail, but the bottom line is, does it work in planta actually? You know? So what you can see here is that if you put botrytis on tomato, you see the growth of the fungus here actually. But if you put the chemical plus the botrytis, it cannot actually grow. Okay? And we can stain with the stain to show that there is a death when there is no chemical, but there is no death when there is a chemical here actually. And if you test it on rose, so botrytis is a major problem in rose industry because when the rose is transported, botrytis can actually affect the rose condition that it loses the marketability actually. So you can see without the chemical, the fungus can grow, but with the chemical, fungus cannot grow actually. And similarly on tomato fruits with chemical, no growth, strawberries, grapes, tomato leaves, and then this is another fungus called monilinia, which is a problem in apple, which is also restricted by this chemical here actually. So not only it can work in the Solanaceae, but it also works on rice blast. Again, this is a major problem in Asian countries. So in a mock infected plant, you can see the rice blast growing, but when we treat with H4 and then apply the rice blast spores, they barely grow on this condition here actually. So bottom line in the summary here is that we think in our pilot study, ATG4 mediated cleavage of ATG8 is an excellent target to identify modulators of autophagy that could potentially be used as a fungicides to control fungal pathogens in plants actually. So in the last couple of minutes, hopefully I'm not running out of time here. Coming back to autophagy, as I said, it's involved in many things. One is the autophagy is being heavily studied in cancer field actually, because of this reason here. So autophagy basically plays a dual role in cancer actually. So during early cancer, when the cancer is beginning, autophagy functions as a defense actually. So it can actually target the cancer cells and get rid of the cancer cells actually. But if the cancer progresses to the metastasis state, then what cancer cells do is they borrow autophagy to survive themselves actually. So what they do is that they will actually digest material from the healthy cells and then borrow the things to survive. So basically autophagy becomes a pro-cancer. But in the beginning, autophagy is an anti-cancer actually. So this is the reason why people have been interested in taking this approach. So it's actually an issue because here you want to activate autophagy. So if you have an early cancer patient, you want to induce it so that you can get rid of the cancer cells. But if it is late, you want an inhibitor of an autophagy because you want to get rid of autophagy so that the cancer cells starve themselves to death actually. So a lot of groups are working on identifying these targets based on autophagy actually. So since we had the system working for plants, we thought we can actually design the same screen for the animal system. So we purified human ATG4 and also human ATG8, and then screen the library to identify inhibitors actually. So as you can see here, 5C3 completely inhibits the cleavage of the human ATG8 by the human ATG4 actually. But in these studies, the specificity is very important actually, but it's not interfering with the cleavage of any other ATG4 mediated ATG8. For example, this is an Arabidopsis plant ATG4 cleavage of ATG8, it still cleaves. This is a tomato ATG4 cleaving tomato ATG8, it still cleaves. This is the botrytis, and this is the yeast ATG4. So this chemical seems to be very specific to the human ATG4 mediated cleavage of human ATG8, often referred to as LC3 actually, okay? So in the mammalian system, of course, there is a lot of information that we can use. I'm going to not say too much about here, but what we think is happening is that the 5C3 is working as a competitive inhibitor of this protease mediated cleavage of the ATG8. And we think it works at a micromolar concentration actually. So since a lot of things we know in the human field, we know the structure of the human ATG8 tagged on to the human ATG4. And here is an active site of the ATG4. And by modeling, we think that the 5C3 actually binds to an active site and interfere with activation of active site. Hence, it cannot cleave the glycine of the ATG8 actually, okay? So because of that, we can actually make mutations in these active site interaction without having to kill the active site actually. And in fact, when you make those mutations, now you can actually cleave actually. So these mutations actually results in cleavage, indicating that presumably the mode of action for 5C3 is to bind to an active site of the ATG4 protease in the human actually. So of course, we have to do a lot more work, to show that its efficacy in the cell actually. All I showed you so far is in the testing. So we need to find somebody who we can work with. We are trying to find somebody in UC San Francisco to test our drugs actually. But there is, like I said, already there are people who works on drugs towards using autophagy as a process to monitor cancer actually. But the popularly used drugs like rapamycin or sarcoptenin, although it suppresses autophagy, but it also suppresses a lot of things actually. For example, AMPK, which is really needed for a lot of other things through this mTOR pathway actually. But the only one which seemed to be specific so far, which is under clinical trial, is this hydroxychloroquine actually. In fact, this drug was initially identified as a malaria drug because what it does is that it inhibits the lysosomal trafficking from the cytoplasm actually. So what it presumably doing is that it is inhibiting targeting of ATG8 into the lysosome actually, so that it cannot function, autophagy cannot function actually. But whereas 5C3 is a very specific inhibitor of an autophagy because it is specifically inhibiting ATG4 mediated cleavage of ATG8 actually. So we think that some of the drugs we have identified here will be a good one to test in vivo to see how these function actually. So I'm going to end there. Hopefully I gave you the flavor of what my lab does, and a lot of the people shown here, Ugrappa, Nagalakshmi, in fact, Ramesh's wife, Nirmala, and Nagu were classmates, comes from the same campus as I graduated in India, University of Agricultural Sciences in Bangalore, actually. And a lot of these two who worked on autophagy now have their own positions in Montana. Yangliang Zhang, who actually developed the proximity labeling in my lab, is a professor in China. Yuan is a current postdoc working on different projects. Cindy and Neeraj worked on different autophagy products I didn't have time to talk about. And then a lot of the collaborators listed here. Of course, we can do all this basic research because of the fundings, actually. Without these fundings from NSF, NIH, USDA, DARPA, and this private institute, we will not be able to pay the people to do the work, actually. I really thank for the invitation, and hopefully you got the gist of what my group works on. I'm happy to take any questions if you have, actually.
Thank you so much for sharing your knowledge on the topic immunity, autophagy, and beyond, sir. We are very grateful to you. I will read out a few questions from the chat box.
Sure.
There's this one question which says, I would like to know the impact of H4 on the environment.
Yeah, I mean, great question. So remember that we don't have, we've not done any test beyond what I showed you actually. But of course, the next step is going to be testing the effect, not just on environment, but the effect on the feeding. Typical experiments are mice feeding people do to establish the toxicity and all that stuff. But what I can tell you is that this chemical comes from an NIH already established library, and we know what this chemical is. And I think it has a great potential because a lot of things we know about this, that we can use it without we have to do all those experiments. But I can't tell you what it is because we have not published, and then we were trying to patent on this stuff. Yeah.
Yes, sir. There's the next question which says, does autophagy interact with other cell functional pathways?
Yeah, I mean, if you just type autophagy on a Google or in PubMed, you see, it has a lot of connection to different things actually, and that makes it sometimes hard because you need to show specificity, but specificity is determined by these ATG8 proteins which interact with the specific protein actually. So yeah, it has functions in trafficking. It has functions in organellar dynamics. It has functions in various aspects of all type of cells, going from yeast all the way to the human cell.
Thank you so much, sir. Now I would like to welcome Jagadamba ma'am for a vote of thanks.
Yes. Good morning, everybody. It is my privilege to thank Dr. Dinesh Kumar, Professor and Chair, Department of Plant Biology and Genome Center, College of Biological Science, University of California, USA. We are fortunate to have you in this webinar, sir. Thank you for sharing your knowledge in plant diseases, then how immune system responds to pathogen, interaction with chloroplast, cell death control during immunity, autophagosomes and autophagy pathway, autophagy cancer. Thank you for sharing all this knowledge, sir. And then it was a wonderful session. And thank you for enlightening us with your knowledge. And thank you very much, sir.
Thank you.
Thank you, Dinesh sir.
Thank you.
Thank you very much for your wonderful talk and accepting our invitation. And on behalf of participants and management and faculty members and all other industrialists, I extend my sincere thanks to your wonderful talk, sir. Thank you very much.
Thank you very much.
Yeah.
Sir, I would like to thank you once again, and I would also like to thank Jagadamba ma'am for the vote of thanks. Now, I would like to request Alakananda ma'am to welcome Chandru sir.
Next speaker.
Yeah, the next speaker.
It gives me an immense pleasure in introducing the speaker of this session, Dr. Chandrasekhar Chervarmat, Assistant Professor, Immunobiology, College of Veterinary Medicine, Iowa State University, Ames, Iowa. He has done his education with the post-graduation doctoral training in the Vaccine and Infectious Disease Organization, Department of Veterinary Pathology, Canada. His PhD is from the University of Saskatchewan, Canada. His master's and bachelor degree is from the University of Agriculture Science, Bangalore. He has been teaching from 2016 to the present date. He teaches respiratory physiology and histology. His research primary focus is on the cell and molecular mechanisms of lung inflammation due to occupational exposure to swine ban environment. Other interests are host pathogen interactions. His honors and awards are a big list. I would like to read few of them. Pfizer Award, Animal Health Graduation Student Research Award in September 2006. He is a recipient of training grant from Canadian Institute of Health Research. He is a reviewer of peer-reviewed journals. He has received Government of India National Merit Scholarship. He has a credit of more than 20 publications and many more achievements. Sir, we thank you for sparing your valuable time here to deliver your talk in spite of your busy schedule. Once again, I welcome you, sir. May I request you to speak on today's topic, mechanism of respiratory and neuroinflammation due to toxicant exposure. Thank you, sir.
Thank you, madam.
Welcome, sir.
Thank you, sir. I hope everybody can hear me.
Yes, sir.
Okay. So please allow me to share the my screen.
Yes, sir, I've allowed you.
Okay. At the outset, I would like to thank Professor Ramesh and the faculty, staff, and students from this wonderful institute. I actually studied textbooks written by Dr. Ramarao, and I had the privilege of meeting Dr. H. Narasimhaya when he came to the Hebbal Veterinary College when I was a BVSC student. Then I did my master's and I moved to Canada, and then I ended up coming to US. And here in the great institute, Iowa State University, I am an assistant professor and I'm studying respiratory inflammation in people who work in agriculture and animal production industry. So just to begin, I have nothing to disclose, no financial interest with any company. I'm a member of American Thoracic Society, and I received funding from various agencies. The main one is CDC NIOSH. It's the Center for Disease Control and National Institute of Occupational Safety and Health.
Good.
To give you a background on the topic, I want you to focus on various professions such as mining, welding, factory, abattoir, or working in slaughterhouse, medical professions, veterinarians, and farmers. How many of you think that farming is a dangerous profession? Working with plants, working with animals, do you think it is dangerous? Most people think it is very safe. Actually, on the contrary, farming is actually equally dangerous. I'm going to tell you why. With these outlines, I'm going to continue talking. I'm going to talk about the historical background, and then I'll move on to the animal production and various toxicants. Then I'll touch upon the respiratory inflammation and mechanism that we have been studying. Then I'll move on to the role of DAMPs, which are damage-associated molecular patterns, and the mitochondrial dysfunction that we have been studying. And then finally, I'll also explain the effect of exposure on the brain or the mechanism of neuroinflammation. Long back, Olaus Magnus actually, he wrote about how the grain dust can actually damage the internal organs. This is way back in 1555. And then in 1700, Bernardino Ramazzini, he wrote a book. This translates into what is known as diseases of workers. He recommended that the doctor should always ask the patient, what is your profession? Because the profession dictates the exposure, and exposure has a long-term implication. So as the human population started to increase, there was a big demand for protein, which comes mainly from meat. So because there was a demand for meat at a cheaper price, the animal production system changed. So what do I mean by that? Initially, in 1966, 1 million farms, they were housing about 57 million pigs. In 2001, just 80,000 farms are housing 57 million pigs. So what is happening is the number of farms are reduced, but the number of animals in each farm is going up. That means we are switching from backyard farming to the factory style of production. So the agriculture, especially the animal production, is turning into a big industry. So with that, what is happening? We actually wrote a review paper recently, and we reviewed all the health effects. So if you look at this map of United States, here is the New York, just to give you a perspective, and there is Chicago. This is the state of Iowa with the two rivers on each side. And here is the Los Angeles on the other side. So Iowa is the center part of animal production as well as the um, agriculture production such as maize and soybean. So this is also the area where there is a higher prevalence of neurodegenerative diseases such as Parkinson and Alzheimer's. This has been linked to the exposure to agriculture contaminants such as pesticide. So on this map, each of the dot actually represents 20,000 pigs. You can see the number of pigs here. And that also shows how many people work in this industry. So I'm going to now show you a picture of the typical pig farm in United States. During the winter months, the windows are all closed. So people who work inside, they're exposed to the dust, bacteria and bacterial DNA, endotoxin, which we also call as lipopolysaccharide, fungal spores, beta D glucan, pig proteins, mites, ammonia gas, methane, hydrogen sulfide, carbon dioxide, and many, many other. So why this is important is because the pork production in US is a very big industry. It produces about 550,000 jobs. It adds big money to the economy. So you can't really get rid of the industry. So people who work in the industry, they actually suffer from respiratory and other ailments. The concentrated animal feeding operations, which are basically factory farming, they affect the community nearby because of the smell. They affect the workers. They also affect the environment. The air quality, the water quality is also affected. So people who work inside, they suffer from respiratory diseases. In my PhD work, what we did, we actually modeled this exposure using the rat and mouse. We put the rat and mouse inside the pig farm, and we showed that the animals experience lung inflammation or lung injury. They also experience what is known as change in airway reactivity. What does that mean? So when you expose them to an agent called methacholine, their lungs restrict. They feel like they are asthmatic. That is the effect of exposure to the barn. At this point of time, the molecular mechanisms were incompletely understood. Because we measured a large amount of endotoxin or lipopolysaccharide, we began to think that that is probably the reason that people are experiencing lung injury. And recently in my lab, what we did with curiosity, we looked at normal human bronchial epithelial cells, the cells from the human lung, and we exposed them to cell culture media control, or we exposed them to the organic dust extract from the barn or the farm. So with exposure, what happened? The mRNA levels of ACE2 protein went up. This is the ACE2, this is the receptor for COVID-19 virus. So now you can understand the importance of working in the barn and the importance of understanding these respiratory diseases, right? So people who work inside the farm, they have lung injury, they are more prone for coronavirus, and they are also prone for influenza virus, which also affects pigs and humans. It can go from pig to human, human to pig. So with this background, now I'm going to talk about our long-term goals. Our long-term goals in our lab include understanding the mechanisms of respiratory and neuroinflammation. And also we want to design the approaches to block that inflammation. These are the two aims in our lab. So we looked at the dust. This dust is really, really fine. It is almost you cannot see it when you are inside the farm. It is very fine dust. When you look under electron microscopy, this is the scanning electron microscopy, you can see the particle size varies from anywhere from 1 up to 40 micron. So why the size is important? That is because these size makes a difference for their deposition. If the particles are bigger, you can actually sneeze them out or you can block them. They don't enter your body. The particle size is around 30 micron, they end up in your throat, you can actually spit them out. If they are really small, smaller than 10 micron, they go deep inside your lung and they stay there and they cause inflammation. So our farmers who work in the field, especially in India, they have a very innovative way of preventing bigger particle. How? I'm going to show you a picture here. So in Rajasthan and in North India, they have a fair called Pushkar Mela. This is a camel fair, and every year, I think it happens in October. It goes for several days, and farmers are sitting here. They're trying to either buy or sell camel. They sometimes get bored and they have a lot of competition. One such competition is for who has the biggest mustache, and he was one of the winner recently. The winners are usually very popular. Foreign visitors come and take pictures with them, and we used to joke that whoever has thick mustache can actually prevent the big dust particles. I don't know whether this is 100% true. I thought of showing this particular style of our farmers, which might be helping them. So jokes apart, now I'm going to focus on the complexity of the immune system, particularly in the lung. So why lung is important? Because on one side, lung is in contact with air. Every minute we are breathing 12 to 14 times. Each breath we are taking in about half a liter of air, and we are leaving half a liter of air outside. So we are in contact with air, and lung is also contact with blood because we are doing gas exchange. We are taking in oxygen and we are exhaling carbon dioxide. That means we are doing that exchange with blood. So anything in air, anything in blood actually affect the lung. Lung also has many cells and soluble factors. Again, the dust in the barn is very complex. It is loaded with different microbial component. Therefore, the inflammation is going to be very complex. We also observed that sometimes there is inflammation, sometimes there is no inflammation. Even in the absence of inflammation, we saw that the lung was sensitized. Lungs were behaving like asthmatic. So it was very difficult to understand because there are too many different damage associated molecular patterns. We began to associate that these damage associated molecular patterns have a role in the dust induced inflammation. So our immediate attention was on a prototype damage associated molecular pattern known as HMGB1 protein. This is a very essential host protein normally present in the nucleus, and this protein, when it comes out of nucleus into the cytoplasm, it has a pathogenic role. So this HMGB1 is the protein that I began to investigate when I started my lab in 2016. When it comes out into the cytoplasm, then it moves into extracellular space. It actually behaves as a potent cytokine. And it is observed that in sepsis and other disease conditions, the levels go up. This HMGB1 can combine with the different receptors known as toll-like receptor 2, 4, 7, 9, and RAISE. RAISE stands for receptor for advanced glycation end product. So by combining with these receptors, HMGB1 can actually amplify the inflammation. Therefore, it is very important to understand its role in lung inflammation. So I hope I gave you nice background as to why we are looking at HMGB1. So people who work inside the barn, they are feeding the pigs, they are caring for them, they get exposed to the dust. And this dust actually activates and starts inflammation in the cells lining the lung or the trachea. These cells actually, they push the nuclear HMGB1 out into the cytoplasm or into the extracellular space. Once it is outside, it is going to induce signaling and it is going to cause more inflammation. And sometimes this inflammation affects the intracellular organelles such as mitochondria. Mitochondria are also very important. They are involved in respiration, immunity, and they also have their own DNA known as mitochondrial DNA. We'll get to that in a minute, because whenever there is damage to the mitochondria, the mitochondrial DNA comes out. And that again amplifies the inflammation. So with this background, now I'm going to show you what are the opportunities to block inflammation. So if you observe the cell signaling due to HMGB1, it combines with RAISE, and then again they induce the signaling intracellularly. So we have opportunity to block HMGB1, or we can block rays. We can also add soluble rays, or we can knock down rays receptor, or we can block the intracellular signaling molecules which are in succession in order to prevent the inflammation and excessive inflammation. That is the goal. So we have these opportunities. So keeping this knowledge in mind, what we did, we designed an experiment. We first took bronchial epithelial cell from human. We exposed them to media, or we exposed them to organic dust extract. And then we used a chemical known as ethyl pyruvate. This prevents the movement of HMGB1 protein from normal nuclear location to the cytoplasm or to the exterior. So we use that as a drug, and we collected cells at different time points. We also did another approach wherein we used the anti-HMGB1 neutralizing antibodies. These antibodies, following exposure, what they do, if there is any HMGB1 that is coming out into the extracellular space, it will neutralize. So with these two approaches, we conducted this study, and what we found to our surprise was the control cells, they have HMGB1 in the nucleus. We have red colored for HMGB1, and then we use the DAPI staining to highlight the nucleus, normal nucleus. You can see HMGB1 is staying in the nucleus. But when we expose to the organic dust extract, the HMGB1 moves from nuclear to the cytoplasmic location. You see here, it is come out into the cytoplasm. But when you treat with ethyl pyruvate, it is again restricted to the nucleus alone. So this shows that ethyl pyruvate is actually very effective in controlling the or restricting the HMGB1. So with that, actually, we started using this in our studies. Next, what we did, we actually used EP, ethyl pyruvate, or anti-neutralizing antibodies. What we found was organic dust exposure produced pro-inflammatory cytokines such as TNF-alpha and IL-1 beta, whereas ethyl pyruvate, it reduced the exposure-induced pro-inflammatory cytokines by preventing the movement of HMGB1 from nucleus to cytoplasm. It actually increased interleukin-10, which is an anti-inflammatory cytokine. So it is helpful, right? Again, the neutralizing antibodies, they also decreased IL-6, which is a pro-inflammatory cytokine. They increased IL-10. So again, these approaches found to be helpful. So next, what we did, we took these results and we moved into an animal model. We actually used the C57 black 6 mice. We gave intranasal saline or organic dust extract for 5 days. And then we gave them either a vehicle or anti-neutralizing antibodies. So we collected various tissues, and I'm just going to show you key results here. What we found was when we measured wet to dry lung ratio, this is a test for edema formation in the lung. So what we found was with organic dust exposure, there was increase in edema, whereas anti-neutralizing antibodies for HMGB1, they significantly reduce the edema in the lung. You all know now what edema does to the lung, because everybody have heard about people suffering from lack of oxygen because of edema in the lung due to COVID, right? So reduction in edema is good for increasing oxygenation. We also looked at inflammatory cells coming into the lung by washing the lung, what we call as bronchoalveolar lavaging. So we found that actually the anti-HMGB1 antibodies reduced the total leukocytes. They also reduced the number of macrophages and number of neutrophils. From a past study, we also had seen that exposure for 20 days actually increased the number of mucus producing cells in the lung. The increase in mucus coupled with edema actually is very bad for oxygenation. So people who work in the barn for several years actually develop edema. They also develop increased number of mucus cells, and they have difficulty in breathing. We also found out that using a test, we measured the epithelial barrier integrity. What it means is the leakiness of the lung epithelium. So with exposure, there is a leakiness of the epithelium which is an increase here. And then with the anti-HMGB1 antibody, we reduced that leakiness. That means we made the lungs more healthy. So with this, we found that HMGB1 is a very good target for reducing the dust exposure induced lung damage. So now we wanted to understand other roles of HMGB1. So HMGB1 has several roles. I'm not going to go into all the details. We actually wanted to understand what is the significance of this intracellular role of HMGB1. Why is it present in every nucleated cell? That is the question we had in our mind. So in order to address that question, what we did, there was already a report showing that global knockdown of this HMGB1 gene was fatal. That means if you knock down this gene, the mice will die. So we decided to target this HMGB1 in particular cells in the lung. So we targeted the cell known as club cells. These are non-ciliated cells in the bronchiole, and they are very important because they work like a stem cell. They multiply and they help in healing the lungs. They also produce a protein known as club cell secretory protein. So we targeted this cell so that we can delete HMGB1 in this particular important cell. So we looked for collaborators and we got the flox mice and cream mice. We bred them. We got the club cell specific knockout of HMGB1. F1 generation of these two mice, when we bred them, we got these and they were quite healthy, and we proceeded with the experiment. So what we did, we went back to the same C57 black 6 mice. We did the similar experiment, exposing them to saline or organic dust or the standard LPS. We exposed for 5 days and we collected tissues. So when we looked at the cellular edema, to our surprise, what happened was, when you expose the animals, the wild type animals with exposure had higher edema, and when you knock down HMGB1, it became even worse. This was our surprise. And also, when we looked at the inflammatory markers such as inflammatory cells, to our surprise, when you knock down HMGB1, the cell numbers were coming down. And we also measured the epithelial barrier integrity. The epithelial barrier increased at the baseline itself because of lack of HMGB1. This was again surprising. So we actually understood that this HMGB1 has a major role to play. Knocking down this HMGB1 can be a double-edged sword, whereas neutralizing the pathologic HMGB1 could be more beneficial. So we are still working on this area to understand what would be the best approach. So with these results, now I'm taking you to the summary. So HMGB1 is known to exacerbate the dust-induced inflammation. Ethyl pyruvate blocks the translocation from the nucleus to cytoplasm and reduces inflammation. When you use antibodies and neutralize, it is beneficial. Club cell specific role of HMGB1, we are still investigating, and at this stage, we believe that it's a double-edged sword. So now I'm going to switch gears and talk about neuroinflammation. So you might ask me a question, why are you moving into neuroinflammation? The reason is simple, because any inhaled particle, we call them PM, particulate matter, they enter through the nose. They use the olfactory epithelium. Sometimes they go to the brain. Here is the olfactory bulb of the brain. Sometimes they enter the lung, and lung produces cytokines. These cytokines, sometimes they come back through circulation and they get into the brain and they cause activation of brain immune cells. So that is why any exposure to contaminant, we should also look at neuroinflammation. So some basics of neuroinflammation. Generally, brain is protected from circulation with the blood-brain barrier, with the exception of some small area. That means normally anything in the blood does not reach the brain. It is good and bad because sometimes you cannot treat certain brain cancers. So the main immune cell in the brain is called as microglial cells. As Dr. Prasad Gabita mentioned, these are the macrophages that you find in other organs. They migrate from bone marrow and they settle in the brain. and they get activated with any inflammation. So you might remember in our village side, people say, now the fear has gone to the brain. They say, Jwara teleg hogide anta ಹೇಳ್ತಾರೆ. That means now the infection has entered brain, right? So microglial cells are very important. In this cartoon, you can see the inflammatory cascade and various effect that it can have. For example, if there is inflammation, it can cause lack of or dysregulated motor control, and it can cause depression and other issues. So the toxicants that we inhale or we come in contact, they also induce oxidative stress. We are going to talk about that. Eventually, there will be structural changes, sensory and motor deficit. With this background, now I'm going to take you into our exposure model. So because we understood that microglial cells are important, initially we turned to microglial cell. Here is a microscopy image of a normal microglia under steady state, control state. If you expose it to dust, they become activated. They start producing bigger processes and they start secreting pro-inflammatory molecules. With this in mind, what we did, we exposed the microglial cells, mouse microglial cell, to the media, and we also exposed them to organic dust extract for 6, 24, and 48 hours. And we stained for IBA1, which is a marker of activated microglia. We treated them with ethyl pyruvate or mitoaposinin. Ethyl pyruvate blocks the movement of HMGB1 from nucleus to cytoplasm, whereas mitoaposinin, it inhibits the cytoplasmic NOX2 enzyme, and it is targeted towards the mitochondria. So with these two approaches, we found that the microglial activation due to exposure can be reduced. That's what we observed. So then we decided to target HMGB1 because blockage of HMGB1 translocation was helpful. Now we decided to use siRNA and block the HMGB1 mRNA. So that's what we did here. With that approach, what we found was the exposure-induced reactive oxygen species, reactive nitrogen species, TNF-alpha, and IL-6 levels actually came down. So knocking down HMGB1 reduces neuroinflammation. That is the take-home message on this slide. Next, what we did, we actually wanted to understand what is happening to the cellular organelle in these microglial cells. So we found out that the exposure activated these cells, whereas NOX2 inhibition reduced that activation. And next, what we did, we took the advantage of electron microscopy. We looked at the control cells. Here is a mitochondria. You can see a mitochondria with cristae inside. Mitochondria is labeled with M. The arrow shows the cristae. Cristae is where the cellular respiration happens. When you expose them to organic dust extract, the mitochondrial cristae are getting disintegrated. The endoplasmic reticulum are getting disintegrated. You can see endoplasmic reticulum are quite healthy here. And also the size of the calcium sequestration bodies, which you see here, they are increasing in organic dust exposure. When you treat the cells with mitoaposinin, you are able to rescue some of these features. And we also found out that there was increase in ER damage. I'm not showing all the data here. And cytoplasmic NOX2 inhibition with MA, we were able to rescue some of the damages. With this data, what we did next was we used an assay called Seahorse assay. We measured various aspects of mitochondrial respiration. We found out that organic dust exposure decreases them, whereas mitoaposinin or MA treatment actually improves mitochondrial function. Then we started to think more into the what other damages might be happening to mitochondria. We knew that any stress of mitochondria would lead to leakage of mitochondrial DNA into the cytosol. So we actually used the mitochondrial DNA specific marker. We measured the expression compared to control. The mitochondrial DNA levels in the cytosol where we had removed any mitochondria, the levels went up. And then when we treated with mitopocyanin, the level significantly came down. So you might ask me a question here. Why are you worried about mitochondrial DNA? Because mitochondrial DNA structure is similar to bacterial DNA, though it is inside the host cell or mammalian cell. Or so we have to worry about it because it increases the inflammation. So what does it do? It actually combines with the toll-like receptor 9, or it also activates the C-gas-STING pathway to increase the production of interferon production. So next what we did, we looked at the C-gas and STING expression. They were going up. And then we looked at the downstream signaling molecules, IRF3 and interferon beta. Those were going up. When we treat with mitopocyanin, those levels were coming down. So this was a good that we can use mitopocyanin to bring it down. And at this stage, we also decided to describe the role of one of these molecules. So what we did next was we actually targeted STING. We used an siRNA and we knocked down the STING. We used a combination of siRNA. With the STING knockdown, what happened? The organic dust exposure induced IBA1, which indicated microglial activation, actually came down. The downstream molecule IRF3 and interferon beta, their levels came down to indicate that you can actually target STING, which is a pathway, a molecule in the pathway activated by mitochondrial DNA. So these are some of the targets we are pursuing now to reduce dust-induced neuroinflammation. Next, we turn to the brain slice culture model. So I'm going to briefly tell you what the brain slice culture is. We take the young mouse and we euthanize them humanly, and we section the mouse brain into very thin, about 200 micron or so. We allow them to heal from that initial injury due to sectioning, and then we either expose them to media or organic dust extract, and then we introduce various interventions. This is basically a mini lung in a petri dish. You can keep them for almost 2 weeks. They have all the regions that you see in a whole brain, and this gives a very easy in vitro assay that mimics the in vivo system. So in this model, what we found was we stained the NUN marker, which indicates the number of neurons. Green color indicates whether microglia is activated or not. So what we found was the exposure actually reduced number of neurons and then activated these microglial cells. It also led to an increase in TNF alpha as well as IL-6 level, whereas mitopocyanin rescued some of these inflammatory markers. So next what we did, we turned to the mouse model of organic dust exposure. So we took the mouse and intranasally we gave the dust with or without oral mitopocyanin, which had already shown activity against the inflammation. So what we found was we also used positive control to see how the dust is traveling. We used the fluorescent LPS marker. So fluorescent LPS marker actually showed fluorescence in the turbinate bones, that is upper respiratory tract, as well as within the lung. And we also found the fluorescence in the olfactory bulb, indicating that anything that we inhale in the workplace, such as pig farm, it eventually ends up in our brain olfactory bulb. We also found that exposure led to the atrophy of the olfactory epithelium. And there was loss of nerve bundle. That means people who work in such environment, they will lose the ability to smell, the olfaction. Then the upper respiratory tract was also damaged. So here in red color, we use acetylated alpha tubulin, which actually stains the cilia in the upper respiratory tract. Cilia in the upper respiratory tract are very important. Again, they help in moving the mucus. There was loss of that with exposure, and there was no rescue with mitopocyanin. That was again interesting to us. We also found that there was increase in mucus producing cells in the upper respiratory tract, and there was some relief with mitopocyanin. When we grossly looked at the whole brain, organic dust exposure led to the congestion of the brain as we looked at them. With these results, we proceeded to do this experiment over 5 weeks. We also conducted neurobehavioral studies. So what we did was we calculated how much time they were able to hold on to the rotating rod. We call it rotor rod. So with 5-week exposure, the ability to hang on to the rotating rod was decreasing, to indicate that these mice were losing the motor control. Their grip was losing. When you treat them with mitoposinin, they were gaining back that ability. That's one thing. We also used a novel scent test, which we call olfaction. So with exposure, the animals were losing the ability to smell. In fact, mice are much better than humans in picking up the smell, but these animals were losing that ability to smell. And with mitoposinin, they were able to gain that function back. The third test was called open field test. This is the test where the animals start to explore the new area. Generally, mice are very inquisitive. They never stay in one place. They start to go into different area. So with exposure, it decreased, whereas mitoposinin treatment, it came back up. It rescued that. And again, with 5-week exposure, there was decrease in body weight. So you can see that exposure caused inflammation in the lung. Exposure led to inflammation in the brain. Exposure also caused compromised motor ability, olfaction, ability to explore new areas. This indicates somewhat anxiety in these animals, loss of body weight. So some of these were completely rescued by mitoposinin. So we were again interested in understanding this. So what we did from these animals, we isolated a pure population of microglial cell. We constructed RNA library, we sequenced it, and what we found was that the exposure led to a significant increase in certain genes, decrease in certain genes. Mitoposinin actually was turning down most of those genes that were upregulated with exposure. This was quite interesting. Again, we validated these results with RT-PCR. These are the top genes that came out, and now we are pursuing some of these as the target. So with this, I summarize that organic dust exposure leads to respiratory and neuroinflammation. Targeting HMGB1 is beneficial. Exposure leads to microglial activation as well as mitochondrial dysfunction. The exposure induces neuroinflammation and leads to degeneration of some of the brain areas. Mitochondrial NOX2 inhibition or mitochondrial DNA signaling could be targeted. Exposure also induces sensory and motor deficits. And we are pursuing some of these genes now, so I don't have much results from those. They are all in infancy. With this, I close. I would like to thank again various collaborators and funding agencies. 5 years ago, when I started my lab, Shrikant was a postdoc in my lab. He has moved on to a faculty job. Sanjana is a biomedical engineer from Bangalore. She completed PhD in April and moved on to her next career. Dhanusha is a veterinarian from Nepal. She's still working. She's graduating soon. Nigel is a veterinarian. He's going to finish in December. This was the picture taken before COVID. So here is a picture after COVID. So thank you. If you have any question, you can ask me now or you can shoot me an email. Thank you.
Thank you, sir. We have a few questions in the chat box. The first question is from Prasad, sir. Whether is your lab, is it have anti-HMGB1 small molecular inhibitor to treat ODE exposed lung damage in mice?
That's a very good question. Right now, what we have, we have one collaborator who supplied us with the specific neutralizing antibody, and we are open for collaborators to provide us with small molecules. And we have actually tried knocking down with the CRISPR. What happens is when you use CRISPR, the cells begin to die. So that is not a good approach. So neutralizing or preventing the movement of this essential protein from nucleus to cytoplasm or cytoplasm to extracellular is very important. So we are open for those ideas. Thank you very much.
Yes, sir. The next question is, does ODE has an impact on muscle action as it is affecting ER?
That's a wonderful question. One of my colleague is looking into that. She's an MD in Nebraska state of United States. She's actually looking into the bone loss, and I collaborate with her. The people who work inside the farm, they always complain of skeletal problem, musculoskeletal problem, tiredness. So mice, when accidentally they did some micro CT scanning, so they found that exposure leads to loss of bone density, and IL-6 is actually involved in it. So because she is pursuing, I am staying away from that area. We collaborate on that area. In general, we collaborate. Yeah.
Okay, so next question is, if the HMGB1 is pushed to the extracellular fluids like lymph cavities, will it affect body's immune system?
That's a wonderful question. I think HMGB1 levels in the circulation actually indicate what is going to happen with the patient. For example, lymph or, for example, cerebrospinal fluid or blood. When HMGB levels go up, it indicates an active inflammation. So any inflammation in the body is not good. For example, in case of COVID-19 patient, they are measuring C-reactive protein, which is another molecule that they pay attention. They are looking at D-dimer values, right, to measure the blood coagulation and all these things. So HMGB1 levels are very important because they keep adding inflammation cycle. Even though the original virus or bacteria has already left the body, these players will continue to cause inflammation, and they need to be neutralized or dealt with some way. They are very essential. You cannot remove them from body. You have to keep them under control. Sorry, I cannot hear you.
Spurti?
Yes, sir.
Yeah.
The next question is again from Prasad, sir. He asks if the brain slice culture model that you have had, is it an organotypic culture model?
Yes, that is correct. I worked with my senior colleague, Dr. Anumanta Kantaswamy. He's a leader in Parkinson area. So I worked with him. We developed that model. It's organotypic culture. You have to work with the specific age of the mouse. We call them mouse pups. And you have to be very meticulous in preparing those. And you have to allow them to heal from the initial sectioning injury. And then you have to work with them. You have to understand what is the original injury that happens when you section them with a blade. Okay, thank you.
Okay, sir. There are last two questions since we are exact running on time. So I'll just wind up with the last two questions. Will the similar kind of mitochondrial damage be seen in urban workers also who are not exposed to organic dust but are exposed to pollutants like soot, industrial dust, construction dust, etc.?
Exactly. That's the whole point. I can give you an example of people who smoke cigarette. I've seen data from vehicular traffic related pollution, ozone exposure, um, toxic metal exposure, even in Parkinson, Alzheimer's. Mitochondrial dysfunction is a very big thing. People are targeting this because if the mitochondria are not working very well, the body becomes sluggish, the cells become sluggish, the innate immune system does not work very well. And then what happens? Mitochondria can be compared to the nuclear powerhouse. They produce ATP, they produce fuel to our body. At the same time, the meltdown of the nuclear power station, what happens? It is not good. It is going to release the toxic product. So mitochondrial DNA and other elements that come out of mitochondria actually affect our own body cells. So it is very common in many diseases to get affected. And people have been trying various approaches to repair them and replace mitochondria.
Thank you, sir. One last question. Can inflammation be there in any part of respiratory tract? If it can be there, then can there be an inflammation in epiglottis?
Yes, that's a wonderful question. In fact, many singers, pop singers, they sometimes bleed in epiglottis because of too much strain. They bleed in vocal cord actually. So sometime inflammation can happen in epiglottis. Generally what happens, depending on the particle size, the smaller the particle, they settle deep down in the lungs, in alveoli, and they are the ones difficult to get rid, and they cause particle overload and stress, you know, yeah, cellular oxidative stress. So anything that is here in throat, you can always spit it out. And you might have seen people, chronic smokers, they constantly try to throw mucus from their throat. So
Thank you, sir.
welcome.
Now I would like to invite my friend Sushma Ballal to deliver the vote of thanks to Chandra.
Sushma, Prasad sir wanted to ask something.
No, just wanted to tell Chandru it was a fantastic talk. I really enjoyed it, and actually, I'm writing you an email right now.
Thank you.
Look forward to catching up with you.
Yeah, thank you so much.
Very good talk. Thank you.
Thank you.
Okay, Spurti, am I audible?
Yeah.
Dinesh Kumar sir wanted to speak, sir.
No. Yeah, all the speakers were great. Actually, I learned quite a bit from the other two speakers.
I extend my sincere thanks to all the 3 resource persons from United States, sir. 3 of them delivered very good talk, accepting our invitation and doing a good talk, sir. Thank you very much, sir.
Thank you.
Sir, Sushma is there. She'll give a vote of thanks.
Sushma, good.
Yes, sir. Am I audible?
Yeah.
Yeah.
Okay. So knowledge shared is knowledge squared. Good morning to everyone. I'm Sushma Ballal, final year student of BSc CBZ from National College, Baswangudi, Bangalore. I'm here to present the vote of thanks for today's webinar. First of all, I would like to thank our principal, Dr. C.B. Annapurnama, and then I would like to thank our guest webinar speaker, Mr. Chandru, for enlightening us with his knowledge. Thank you so much, sir, for giving us a brief account on these things like historical background and animal production and toxicants, role of DAMPs and mitochondrial dysfunction, and exposure-induced neuroinflammation. I would like to thank you for giving us such an informative and knowledge-illuminative talk, sir. And then I would further share my hearty gratitude to Professor Ramesh PL, HOD of Department of Zoology, and Professor Shakuntala L and Harish sir for giving us this wonderful opportunity to attend this amazing session. I would like to thank all the participants as well for being so patient and gaining yourself the knowledge that could be used in your further upcoming studies and your professional field as well. Thank you, sir.
Thank you, Sushma. Yes, Spurti.
Yes, sir. Now we will have a 25-minute short break. So we will meet back again at 11 o'clock exactly. So it's a quick breakfast break. So yeah, we will meet back at 11 o'clock. Then we will have 3 more speakers. That is Dr. Viresh P. Virapur speaking on impacts of toxicants on human health. Dr. G. Ravikant speaking on animal, planet, environment, and human health, need for One Health program. And lastly, Dr. S. Vijay Kumar sir, toxicology research in animal models. We are eagerly waiting for these topics as well. And I am also grateful for the topics and speakers who have shared their immense valuable knowledge to us before. Thank you all once again. We'll meet you all back.
Thank you, sir. Thanks to Prasad sir, Dinesh sir, and Chandru sir for wonderful morning session for us and wonderful afternoon session for you people. Thank you, sir.
Dr. Ramesh, I'm going to take a break.
Yes, sir.
It's 12 o'clock here, midnight. So
Yeah. That's it. Yeah. Thank you, sir.
Yeah. I'll also be taking leave. Thank you. Thank you, Spurti.
Yeah.
You did a fantastic job. Thank you.
Yeah.
Thank you, sir.
Yeah.
Thank you, Professor Ramesh.
Yeah, thank you, sir. We have to thank for you people.
Yes, and I also enjoyed your talk, Dr. Dinesh.
Yeah.
So good luck for the next 3 talks. I won't be there actually. Thank you very much.
So in coming days, you are using your resources for conducting another webinars.
Oh yeah, sure.
We are connecting. Okay, sir. Thank you, sir. Spurti, can we
Yes, sir. I'm doing it now. I'm sharing the brochure now.
yeah, sure.
Just a second.
Because ಅವ್ರಿಗೆ 12 o'clock ಹಾಕಿದೆ ರಾತ್ರಿ ಅವ್ರೇನು ನಾವು ಸೆಷನ್ ಹಾಕಿದ್ರೆ ಹಂಗೆ ಕಂಟಿನ್ಯೂ ಆಗಿರೋರೇನೋ
Sir, are you able to see the screen?
Yeah, it is visible. I think it is only 10 minutes Purti, not half an hour. It is already 10:45. I think you come back after 10 minutes.
11 o'clock, sir. I've mentioned it as now 11 o'clock.
11, let it be, but we'll come back at early 10 minutes.
Okay, sir. Requesting all to please mute your microphones. We are still in the session, but then we have just taken a short break. We will remove the contestants whom we see who are, unmuting their microphones. So please mute the microphones. Thank you.
சத்தியவேல் சத்தியவேலுக்கு சொன்ன ஹோல்சேல் கடை வச்சிருக்காருல அவங்க சொந்தக்காரங்க. ஆமா ஆமா.
Hmm.
The function of macrophages are affected.
Hello, Ramesh sir. Hello. Anybody here?
Yes, ma'am. It's audible.
Yeah. Madam. Okay. Welcome. Good morning. ಎಲ್ಲ ಇದ್ದಾರೆ ಪಾರ್ಟಿ ಸ್ಫೂರ್ತಿ ಸ್ಫೂರ್ತಿ
Yes, sir. One nimsha, sir. I'll stop screen sharing.
Yeah, you can stop and request the participant to rejoin.
Okay, sir. 1 minute.
Almost 240 250 people are there, but I think some of them are left.
Nearly 130 people left the meet, sir.
No.
When it started, around like 500 is there. Spurti madam idara?
Yes, sir. 1 minute, sir.
Spurti, we are, sir.
Sir.
Now we can start, sir. I think in another 2 hours. Yeah.
Ramesh avare.
Yeah, tell me, madam.
Share option ಕೊಡಿ sir. Biodata screen ಮೇಲೆ ಬಿಟ್ಟಿರ್ತಾನೆ.
ಯಾರು ಹೌದಾ?
ಅವರು ಸ್ಫೂರ್ತಿ ಅವರು ಕೊಡಬೇಕು.
Okay. Good morning, sir. Ravi sir.
Good morning, sir.
Morning.
Morning.
So you attended the previous sessions?
Yes, sir. I attended Chandru talk and then partly attended Dinesh and yeah. No, Prakash also was there in the beginning.
Okay.
Yeah.
Sir, namaste.
Namaste, sir.
Yeah, namaste.
Spurti?
Yes, ma'am. You can start. Actually, we were waiting for 2 more minutes, ma'am.
Okay.
No. Okay.
Spurti avare.
ಅಲ್ಲ ಈ ನನಗೆ ಸ್ಕ್ರೀನ್ ಶೇರ್ ಆಪ್ಷನ್ ಕೊಡು ಒಂದೇ.
ಎಸ್ ಮ್ಯಾಮ್, co-host ಮಾಡಿದೀನಿ ನಿಮ್ಗೆ. Everyone else, can you please mute your microphones? We request everyone else to please mute your microphones.
Yeah.
ವೈಸ್ ಚೇರ್ಮನ್ ನೀವು ಸ್ಕ್ರೀನ್ ಶೇರ್ ಮಾಡ್ಬೋದು ಇವಾಗ.
ಆಯ್ತು ಮಾತಾಡ್ತಾ ಎಲ್ಲ ಶುರು ಮಾಡುವಂತೆ ನೀವು ಸಿಗ್ನಲ್ ಕೊಟ್ಟ ಮೇಲೆ.
Morning sir. Good morning Veeresh sir.
Good morning sir. Good morning everybody.
Yeah, good morning.
Can you make a co-host so that I can share once immediately?
No sir, just 1 minute sir.
Okay, thank you.
Yeah.
Sir, can we start?
Yeah, Spurti. Yes.
Or are we waiting for a few more minutes?
No, you can start.
Okay, welcome back everyone. We will now resume our webinar session. Please mute your microphones as it will cause a lot of disturbance to the speaker. So now I would like to welcome Vice Kamala ma'am to welcome Veeresh sir. Welcome ma'am.
Very nice of you, Spurti. It's my pleasure to introduce Mr. Veeresh sir for this August.
Reliable. So according to CDC's
we request you to kindly mute your microphones, please.
mortality is almost 54%. So if
Please.
Can all of you see?
Yeah, no ma'am, not yet.
Screen ಕಾಣಿಸ್ತಿಲ್ವಾ? ಬನ್ನಿ.
ನೀವು ಕಾಣಿಸ್ತೀರಾ ಮ್ಯಾಮ್, ನೀವು ಸ್ಕ್ರೀನ್ ಶೇರ್ ಮಾಡಿ.
ಮಾಡಿದೆ.
No, ಹಂಗೆ ಹೇಳಿಬಿಡಿ ಮೇಡಮ್. You can tell.
I just want people to.
Okay.
Okay, hope you can see now.
Yeah.
So it's our pleasure to have Mr. Veeresh Prabhakar Veerapur sir with us. Mr. Veeresh is a professor at Sri Siddhaganga College of Pharmacy, Tumkur. Mr. Veeresh had his Bachelor of Pharmacy from Karnataka University, Dharwad, and further completed his M.Pharma from Pharmaceutical Chemistry and PhD from Manipal College of Pharmaceutical Sciences. So Mr. Veeresh has in total of 20 years of experience and 81 research papers to his credit. It is not a small achievement. In 20 years, 81 papers is very, very huge in number. So next, his impact factor. See, this is a gravity. So in any researcher,
90. Yes. Spurti.
Yes sir.
Yeah, mute ಆಗಿದೆ. Okay. Yes. Yes, madam. Go ahead.
ಕೇಳಿಸ್ತಿದೆಯಾ?
Yes. Audible.
Okay, very nice. Sorry. So
Excuse me, there is
the people. I do it. So the gravity of the scientist is always in his impact factor and the H index. H index is 28, and he has co-authored 4 book chapters, and he has received 6 research grants worth 80.6 lakhs. For any college teacher teaching in a institution, this is a very good amount. He established research collaboration with industries and academic universities in India and abroad. So USA and Togo, West Africa, and other countries. Mr. Veerapur has received several awards, including AICTE Career Award for Young Teachers in 2010 itself, APTI Young Pharmacy Research Teacher Award in 2011, Stars Award for Teachers, in that Award for Excellence in Academic Research in the year 2012. It is given by American Association of Government College of Pharmacy and alumni of the Pharmacy College of the Bangalore who are residing in America. So, and he has also received 1 travel grant, 1 best oral and 8 best post awards during national and international conferences. This is going to be a huge benefit for us. That means he can guide the students. The oral and the poster presentations are the common in any.
Yeah. Yes, madam. Go ahead.
Programs conducted across the country. So now I request Mr. Virap. Impacts of toxicants on human health. So this is our, sir, it is our pleasure to have you here.
Thank you very much, madam.
I'm sorry for a few disturbances in between. So
No problem.
Again, please, over to Mr. Virapur, sir.
Thank you, madam.
Welcome, sir.
Thank you. Good morning, everybody.
Yes.
Thank you very much. Okay.
Ma'am, can you please stop sharing the screen?
Stop sharing, sir, madam. Yeah.
Hopefully, you people are seeing the slides now.
Yes, sir. It is visible. Yeah.
So shall I start, sir?
Yes, sir. You can start.
Okay. Good morning, everybody, once again. Thank you very much. I would like to thank Dr. Ramesh, sir, and his eminent team of National College Bangalore for organizing this very interesting webinar in this particular pandemic. So even though pandemic is there, there are webinars and other things like that. We can increase our knowledge. So now I'm in a different platform. I'm just delivering the talk through this particular webinar. And I also thank the organizers for giving me opportunity to be a part of it and presenting my topic on impacts of toxicants on human health. So it's a very general topic for me. It's not a research topic I'm going to deliver with the audience. So as Madam introduced me about, I'm Dr. Viresh P. Virapu, Professor of Siddhanga College of Pharmacy. I am going doing this particular research on animal research on different toxicants. Maybe you can take an example of diabetes-inducing toxicants or neurotoxicological aspects. We come across, there are so many toxicants which are directly or indirectly affecting the human health. So I would like to start with a thought. Thought says all substances are poisonous or toxicants. There is none that is not poison in world. The right dose differentiates the poison and a remedy is a famous statement from Paracelsus. So what is the take-home message from this is every chemical components we are using day-to-day life, all involved with the chemicals, with a right dose or a right choice of chemicals, we can avoid the toxicant effect of it. The outline of my talk, goes like introduction about my topic, types of toxicants we encounter in day-to-day life. What are the health effects of those things? I'll cover some examples and their remedies to overcome this one and end with a conclusion and statements. So what are toxic chemicals? One minute.
Yes.
So everybody understand the poisonous and toxic substances which have a deleterious effect on a health of a human or animals. So mostly we concerned about most toxicant, we can say they are originated from pollutants, especially polychlorinated biphenyls, PCBs they say, and another one is persistent organic pollutants, which are mainly base of dioxin and their analogs. These toxic chemicals, they not only harms the health of humans or animals, also it affects the environment. There are some examples of toxicants. Everybody knows these statements. So mostly used pesticides, especially for protecting or increasing the crop in agriculture. Okay, so we have to know about these pesticide effects because we are real consumer day-to-day life of these vegetables and other things. So second class of toxicants are pollutants. Already I expand the word of POPs, that is we call it as persistent organic pollutants. They have enormous impact on human health. So later is carcinogens and mutagens, maybe some drugs which also be a resulted into a carcinogenesis and mutagenesis. There are enormous drugs which are teratogenic in nature. Thalidomide is the best example of teratogenic effect of its. Next is allergens. Heavy metals toxicity is also very prime important in this particular list. And other things is food-related toxicants. Day to day, if we consume these things knowingly or unknowingly or ignorantly, I will discuss some of examples of it. And lastly, the household toxicants. So daily life, what all toxicants we are encountering it, how to avoid those things. There are a list of 10 best ranked chemicals which are known to be toxic. Okay, known to be toxic, and they are listed ranked based on the frequency of exposure and to the toxicity to the human. Okay, arsenic, is most frequently used environment, lead, mercury, and polychlorinated biphenyls, which are very toxic in industrial pollutants and all sort of things, and benzene, benzopyrene, okay, smoke. So this is a very carcinogenic toxicants. They are listed in the priority wise based on the frequency of exposure to the humans. So what they especially maybe from industrial pollutants as a disposal or emissions, or maybe a pesticide use or open burning, especially in or strike, what they say, the tire burn, they do all sort of things, and incinerations, PCB I said, and automobile exhaust leads to a very persistent organic pollutants. What exactly persistent organic pollutants means? They are very stable organic compounds. They live for longer period of time in the environment. It may be mixed with water. They are very stable chemical compounds, especially dioxin type of chemicals. They ultimately they circled into in the environment, maybe our animals, plants, soil, and it affects ultimately humans. So what exactly these pollutant aspects will affect is, especially main target is they are nerve damage chemicals, neurotoxic, cardiotoxic. They are lung damages. They lead to enormous number of what you can say cancers. And nowadays people are more known thing is most of the people are affecting with the cancer because of these pollutant toxic compounds. Okay, so this better disturbance is there. Is it okay up to here?
Carry on, sir.
Okay. So, okay. So I'll give some what you can say toxic compounds, their utilization, and what is the impact on health and all sort of things. When you talk about, so first standover is the pesticides, because the farmer spray these pesticides to improve the crop. Okay, to protect the crop, but also it affects to our consumers also. So pesticides, they spray in order to prevent or destroy the particular pests. Pest is not only a creatures which also include rats, mice, and all sort of things. Even though herbicides, we say that they are unwanted plants which are grown in a field like weeds. To remove those things, they'll use it. And don't forget in household to kill the cockroach, because most of the people fear of a cockroach. They keep it the pest controller, the sprayer, cockroach repeller. So that is we should use very carefully. That is also very toxic compound chemicals. So what is the impact of these pesticides on human is they create a disorders of endocrine glands, pancreas, spleen, all sort of things will be damaging. Along with that, they damage kidney and liver. Okay, so mostly they compromise the immune system. That is very important nowadays. Everybody now talk about mostly because of the corona pandemic, people want to increase their immune systems. One of the thing is get rid of these toxic pesticides. Okay, so another thing is what is the impact is that it increases the birth of mentally and physically challenged children. So we have sort of examples where the mentally and physically challenged children are born due to the excessive use of pesticides. Dwindling sperm count is very concerned in a worldwide, because one of the reason I'm talking about, it may be due to a toxic pesticides. So along with the suffering of humans, it is severe deficiency of micronutrient in the soil is also resultant of this pesticide use. Okay, so the traces of chemicals in a banned pesticides have been found in breast milk in many people in Punjab. Take my example where Punjab is a place where most of farmers agriculture field is there, and they use so much of pesticides. So many people in Punjab, they are suffering with cancers. So because of one of the reason is pesticides. Another very dwindling fact is these pests are evolved with the combat effect of pesticides. Therefore, we have to develop more pesticides that resulted into more toxic pesticides. So alarmingly, there is a rise in number of insecticide or pesticide resistance pest. DDT, people used to use, now it is banned. Okay, there is a development of resistance to this DDT, and all sort of things are resulted into this particular one. So how to control this particular impact is create awareness regarding the pesticide, especially to the village people. Awareness. One thing is that even though educated people also are more negligency about these pesticides and impact of it. So overall awareness program has to be there. Already the people are doing it. More efficient laws and strict enforcement to ensure the banned pesticides not to be used. So what is the replacement for this? Use biological pesticides like canola oil or baking soda. This is maybe a very preliminary one. The number of biological pesticides are discovering nowadays to replace these harmful pesticides. Proper research is also needed to discover new pesticides with a less environmental or health impact. With this all environmental and pesticide impact, there is the field itself will started because of this impact of health impact of these toxicant is environmental toxicology. It's a branch of science where multidisciplinary field of science which are concerned about the study of harmful effect of various chemicals, biological, physical agents on a living organisms. Even people are talking about paint, whatever you're using, that is also harmful to the humans. Okay, so the study of these impact, so whatever paints are developed now, whatever drugs which are developed now, they have to be checked the toxicological studies. I think in the next talk, Dr. Vijay Kumar will emphasize on how the toxic chemical are tested. So actually, the global environmental movement was started by seeing the publication of Silent Spring by American marine biologist Lochie Lewis. They understanding based on the development of understanding about the global alarming environmental harmness and human health. So with this, I will have some case studies to emphasize that how these toxicants will impact on human health in different case studies. One of the case studies, maybe whether you heard about this particular herbicide, is called as Agent Orange. It is a herbicide or defoliants to remove the leaves of animals. People use this particular Agent Orange. It is a mixture equal amount of 2,4,5 trichlorophenoxyacetic acid. Okay, it is a trichloro and dichloro phenoxyacetic acid. It is a chemical itself is toxic, which is used as herbicide in 1960, 1950 range here. So one of the biggest contaminant of this particular Agent Orange is TCDD. It is a chemical tetrachloro-dioxane derivative. It's a dioxin, again a dioxin derivative, which is very toxic chemical. So because of this toxic chemical, it affects the human enormously. So why it is called as Agent Orange? Because the name itself is saying the chemical herbicide is orange color. During the Vietnam War, US military sprayed this particular Agent Orange of 757 lakhs of liter of this chemical onto the fields of Vietnam in order to eradicate or remove the vegetation or a forest. Why they done this particular work is to prevent the enemies to shelter among these forests. Therefore, they sprayed it. What is the impact of this spraying in near 1960 range? 4.8 million Vietnamese people are affected with this particular chemical. 4 lakh people are died because of this particular chemical, impact of chemical. 5 lakh children born with defects, birth defects. I think you can see the particular pictures. That was the enormous impact of Agent Orange. So after this particular incidents, people are aware better to ban these Agent Orange, and US different environmental agencies are banned this particular chemicals. The intention is to hide prevent the hiding of enemies, but impact is very enormous. So the impact of dioxin, as I said, it's not only as Agent Orange. So day-to-day life, we burn so much of tires or burning of the trees, all sort of things. I think forest burn also contain derivatives of dioxins, which are very stable, especially this structure. Tetrachloro dioxin derivatives are very harmful to the humans. So I think you hopefully understood the impact of it. And what are the source of it? Volcano eruption, fire, forest, as I said earlier, incineration of plastic, which we do most of the time. Now people are avoiding of it. Paper industry, thermal industry, waste incinerations are the resulted of the dioxin derivatives, which are very stable. They are there in the whole environment. That definitely affects the human health and environment also. So second case study is where endosulfan presidy of Padre region of Kasargod. Earlier it was part of Karnataka, now it is with Kerala, everybody understand. And this particular endosulfan is a chlorinated organic pesticide. They sprayed to have an impact of herbicidal effect. Okay, so the enormous effect of endosulfan is on, especially they driven with a birth of a mentally retarded and people, and their the sexual hormones depletion is resulted of it, and their blood is traces of endosulfan was observed. The water, air of Padre region is still the traces of endosulfans were recovered. And maybe in 10 years back, when I was doing my PhD there, we used to check the endosulfan traces of endosulfan present in the particular samples taken from the Padre region in Kasargod. And the enormous impact of human health is picturized in this particular head pictures. So don't feel it. I'm showing you a lot of photos where the lot of it's just heart-wringing aspect is so much of impact happens. Still, they're born with a deformation, malfunctions of it. This is what the impact of endosulfan sprayed. Actually, endosulfan was sprayed with a helicopter actually. Okay, so the impact of it is still people of Kasargod of that Padre regions are suffering with this particular endosulfan. So next case study is thalidomide residuum. I think this is a drug people used for as a anti-anxiety. If anybody is having anxiety, people prescribe that. They are used for insomnia. If you're not having a sleepless nights, better to take this particular thalidomide, which is introduced in 1957 in West Germany for these particular remedies. Later, they used for to treat morning sickness for a pregnant woman, especially in working women want to go for working. They have suffered from morning sickness. They cannot do it. They attend their job. Therefore, they prescribe this thalidomide to them. Okay. So what was the impact of this particular thalidomide was very drastic. People worldwide will be eye-opened and check it out. What is the impact of it? So all again deformed babies are born to those pregnant women, especially initially 5,000 what I can say children are born with deformations, and their progenies were born with deformations. So after seeing that, people are now later they discover which isomer of this thalidomide which is impacting this particular teratogenic impact is your S isomer. It's a configurational isomer of thalidomide S, which is toxic, which are R thalidomide is not toxic. Based on this, nowadays, people are in a drug discovery, people are checking whether which isomer is toxic and which isomer is safe. Okay, so that emerges chiral separation and chiral synthesis emerges into it. One best example I can give is everybody consumed cetirizine. So maybe 10 years back, cetirizine was there. Now people are talking about L-cetirizine, levocetirizine, levorotatory cetirizine. So the dose of levocetirizine is only 5 mg, whereas cetirizine is 10 mg. And it gives you a drowsiness, the cetirizine, whereas L-cetirizine is not gives you a drowsiness. That part of emergence of looking for a particular isomer form of a drug is entirely credited based on that what I can say thalidomide residue. I thought I'll share with you. So comes to a next aspect is food related toxicants. Okay, and most of the time, knowingly or unknowingly, we will have this. One such thing is unpreserved food. Okay, so unpreserved food and soil also contaminated with the organism, especially anaerobic organism like your gram-positive anaerobic bacteria called Clostridium botulinum. it produces a toxin called botulinum, which is a very neurotoxin. When you type a very toxic chemical, this name will come in a Google, botulinum. Even though it is not sometimes they say it is originated from pathogens, but most of the time in a food, unpreserved food, this particular organism will be there, and it produces a toxin called botulinum, which is very toxic. Next one is to the young generation. Okay, so it become a fashion to us that have a Coca-Cola or any soft drinks. They are estrogenic in nature. They are called as estrogenic mimic. Okay, so they may be color, flavor, sweeteners or additives which are in synthetic in nature, and they give a estrogenic. And the soft drinks, whatever we are using, these are acidic in nature. That particular thing will harm to it. So nowadays people are most of the people are avoiding such soft drinks. They go with going for a coconut water rather than going for a Coca-Cola. Recently football Euro is going on, and Ronaldo, and he removed that particular soft drinks from during his press conference. Emerges a lot of losses to the company. I think you recently heard about that, and Cristiano Ronaldo, and this particular impact, everybody should avoid these soft drinks. And another very important food, which is white in nature, and they are enemies to the human health, that we eat unknowingly, knowingly, or maybe ignorantly. So one such white enemies, few are sugar, okay, salt, and maida. Everybody understand more consumption. Please understand at this juncture, I will emphasize that overeating any what you can say, amruta also, it will be a dangerous. Similarly, sugar, salt, we cannot avoid, but the replacement you can check it out. So what is the replacement of a sugar is? It's a black jaggery. It's not brown or golden jaggery I'm recommending a black jaggeries. Nowadays organic black jaggeries are available. You can replace with it. And salt, you can use a rock salt, which is having a lot of mineral oils than the salt which is derived from a sea. Okay, and coming to a maida impact is how we process, how we get the maida from the wheat. What they do is they process to get the maida. In order to get that, they add a chemical called alloxan. Have you heard? I don't know. So you can Google it, maida, harmful effect of maida. I think it will come. Alloxan is a chemical. We are using those chemical alloxan to induce a diabetes in the animal. So that particular alloxan they are adding to process it. It is one of the chemical which is used to process the formation of maida. So, so hopefully you're understanding. So alloxan induces the diabetes. Therefore, maida contains the traces. More amount of eating of maida will gives you a diabetes. That's why India became second in the incidence of diabetes. So one of the reason is these food old origin what we eat. So better to replace with a whole wheat flour, what you can use, even though people say it contains gluten, which is also toxic. What you can say, ragi and other things are best replacement of it. So better to avoid consumption of these aspects. Apart from this, even your pasteurized milk is also harmful, people say, because pasteurization or heating of a milk can remove the vital ingredient from the milk. Okay, and therefore better to take that particular milk, raw milk, what we say that. So that we observed in our childhood. My grandfather used to drink, so he used to take a milk from the immediately, he used to drink without heating it. That is more useful rather than going for the pasteurized milk. Another reason for the harmful, we say harmful to nature of milk is especially farmer, especially they give chemical called oxytocin to increase the milk production. That is also one of the reason it's a harmful effect of a milk. So nowadays people have started understanding milk without the tea without milk is also better rather than adding a milk into it. So next is white or a refined rice are harmful to it. Unpolished rice are always a better option than a white or refined processed rice. Okay. So one more white, what you can say, enemy to everybody is young generation who take a lot of street food or maybe food which is of gobi manchuri and other fried items. They are contaminated with white chemical crystals, whatever you're seeing on a slide, which is monosodium glutamate. It's also called as China salt because these are the people who use this particular chemical. In India, they call it as Ajinomoto. So Ajinomoto, why they use this Ajinomoto is to improve the flavor as well as taste. Okay. So what is the impact of this particular monosodium? In a small quantity, no problem. But in a continuous consumption of in a high concentration, chronic exposure of this monosodium glutamate is it gives you a neurotoxicity. That means behavioral changes, anxiety, depression. So one of the major reason for chronic consumption of monosodium glutamate is these things. We use this particular monosodium glutamate, we inject to the animal and we check what happens to their brain and what is their behavioral changes in different animal models. So therefore, avoid food which are containing monosodium glutamate, Maggi. Okay, so people say it's banned because of heavy metal content. Another thing is they observe monosodium glutamate with it. So these are the impact of these monosodium glutamate, impact of white enemies to the human. So what I emphasize here is better to avoid chronic exposure to these particular white enemies to the human. So next come to a next toxicants, which are household toxicants, which are routinely we use it. Maybe hopefully you are seeing it. So most of our water filling, we are using these plastic vessels aspects of these plastics are estrogenic in nature. They are in estrogen mimics. That means estrogen, what it does is the hormone which gives you a male will give you a female characteristics. And these plastic is a non-biodegradable and they remain in the earth more than 100 years and they are toxic to human things. And there are a lot of cities which are garbage containing plastics. Okay, nowadays these things are avoiding, but certain places still it is there. So people burn these particular what you can say garbages that resulted into a persistent organic pollutants, as I explained earlier, that resulted into a toxic effects. So now if anybody burn in open air, definitely you will get a fine. So other household thing is, it may be a bleach what we use. Carefully use these things, drain cleaners, carpet cleaners, ammonia, most of the things, foam containing materials, foam aspect. People with a, what you can say, bed with a foam, and that is also gives you a lot of toxic chemicals. which gives you a toxic effects. And fragrance, air's freshener, what we use, that is very toxic. It results into obesity, breast cancers, as well as it gives you allergy related asthma and all sort of things. Another very important thing is perfluorinated compounds are there in non-stick cookware. Okay, that is also a very harmful impact. And whatever plastic bottle we are using, better to use a, what you can say, glass bottle for drinking rather than using a plastic bottles, because they contain a chemical called phthalates and PVC materials, and they have bisphenol A and bisphenol S, which are hormone disruptors, or they inhibit the hormones, leads to many cancers. This is one of the reasons. And children's with toys which are of plastics, which contain this. Nowadays, people are asking for a material which is of free from BPA, that is bisphenol A and bisphenol S. Okay, nowadays they are talking about and avoiding. Another very important thing is, whenever you go to a market and they'll give you a slip with a what you can say, that glossy slip will be materials which contain a bisphenol A. This is very toxic. Okay, so these are the few things about the chemicals, and especially for baby and children's products. Hopefully, which is contaminated with phthalate. And another very important thing is the foams what we use, that is very carcinogenic, and formaldehyde, which is found in baby personal care materials, which is very toxic and neurotoxic in nature. So we have to avoid with this. And what happens to this particular plastic chemical component, which are recycled in the whole environment, and gives you a lot of toxic effects, what you are seeing here. Another very important thing is beauty enhancers. These are also estrogenic mimics. Synthetic chemicals which are used in most of the cosmetics are in toxic in nature. Better to avoid such things. And they contain, especially one of the example is sunscreen, which contain a oxybenzone, which is again a hormone disruptors. Why young generation are giving more puberty is because of in young age, maybe 10 years, 9 years, also they are getting into a puberty. It's because of these particular chemicals which are using it. These has to be avoided. Another very important toxicants to the environment and human health is e-waste. So whatever e-wastes are there, their disposal of these e-waste disposal, they contaminated with the water and air pollutant, and they contaminate the plant. Ultimately, they contaminate humans. What happens with this particular e-wasted chemicals, especially lithium battery and other components which contain more amount of heavy metals, that resulted into a variety of cancers, especially lung cancers. Very small, like vomiting, headache, sleeplessness is another one. Appetite, diarrhea are the more harmful effects of e-waste what you can get. And it also affects neurological effects in humans. Not only it affects humans, it also affects the plants. So ultimately environment. They reduce the crop production, impairment in the metabolism, and reduce the food value is also a impact of lithium battery or e-waste is a resultant of it. Apart from these household impact, very general, I thought this particular slide is very apt for present situations, what we are there in a pandemic. So this particular one is deforestation. When you go for a deforestation, these animals are containing a number of viruses and pathogens. They go and reside into humans. That's why we are getting a coronavirus. Now I will not go on deep of it. So I thought deforestation. Therefore, plantation is also one of the reasons we can avoid these human diseases and toxicants. I think I'm talking about this particular paper slip from maybe 10 to 15 years back. People are talking about, they are allergent to most of household foods and environment. That's what she's taking testing of all the allergent. Okay, so now people are becoming more allergent and started using mask. Okay, that was a slogan. And this was whenever you go for outside, people are using a mask. And now the same situation is happening because of a pandemic of corona. Everybody is wearing a mask whenever they go to a public. So we can't just ignore these problems, whatever we discussed, and most of the things you know about it. Like this, we cannot ignore these problems. 1 minute. Huh? I think this thing is disturbing.
Yeah. No, sir. Now it is okay. Go ahead.
Now it's okay. So we cannot ignore these problems. We have to. What we can do for this problem is right attitude to avoid, especially. So main conclusion of my today's presentation is reduce the waste which is produced. Most of the time you have to avoid, okay, avoid whatever you can reduce it, usage, reuse it, whatever material you are using it, recycle it, whichever is possible, recovery from the waste, you can recover the energy source, what you can do, it's topic of discussion. Otherwise, the safe disposal of a waste is very important. Okay, so these are the few aspects what we can use. The right education about these toxic effects of it, maybe from environmental pollutants or maybe pesticide usage, or it may be of maybe household use of food originated aspects you can avoid wherever is possible. So I will stop my discussion with the Mother Teresa quote, saying that if we have no peace, it is because we have forgotten that we belonging to each other. Nowadays, this statement of Mother Teresa is true because we are not bothered about other people. With that particular statement, I'll give an quote. Earlier, people used to do a business without harming anybody. They have that ethical issues in them. Nowadays, business itself is harming and getting the money more, okay, without bothering. Okay, so I think this statement is very apt to say. So what we can do is together we can change this notion. We should not harm to the environment. In turn, we can prevent the harmness to the humans and animals. So let us care about our environment and creatures. We are all in this universe. And this is what my take-home message of my presentation. Thank you very much for patience.
Thank you, sir. I'll be reading the questions from the question box.
Yes.
What would be the fate of aquatic ecosystem when biological pesticides get accumulated as bioaccumulation?
That is it's a subject of again problem. But if those aquatic animals, that's what I'm saying. So whatever biopesticides, whatever we're using, it should not harm to any ecosystems or environmental aspects. So in that, it can be avoided.
Okay.
Proper testing is needed.
Okay. Next question is, since there's recently eutrophication in River Ganga, how is it going to affect the quality of the water?
The quality of water, that's what the whole process of cleaning Ganga happens, but the traces definitely will be there. So I think you saw that, especially persistent organic pollutants, it's very difficult to remove. One of the biggest problem with these things are, they are very, what I can say, easily can penetrate into a tissue because of their lipid solubility. Okay, and the cleanness of water can be tested and can be avoided.
Okay. Next question is fresh milk free from microorganisms?
Yes, no, they have microorganisms. Even the people say that there are certain microorganisms which are very useful to the human. So why pasteurization or heating of milk is immersed into a day-to-day life is to kill that particular harmful this thing, or you can say microorganisms. Yes, I agree that present situation, definitely the milk, whatever we are getting, which is not a free of harmful microorganisms, as well as the chemical which is what I can say injected to the animal in order to get the more milk production. That's what I said. The business has to be, we humans contaminated the brains so that it gives you a harmful toxicants. That's what I said. Better to avoid even no. See, it is a balance of the good microorganisms and bad microorganisms. Both will be there in a milk.
Okay, sir. Last question is that will pasteurization kill microorganisms?
Definitely, it kills microorganisms. That's what people say. So, in the prospect of good microorganisms they are talking about, we should not pasteurize. But I said in earlier answering to the earlier question, pasteurization kills the harmful also. So presently situation is other techniques we have to use, like freezing. It may be a technique where you can kill some microorganisms. The freezed one we can use rather than going for a pasteurization.
Okay, sir. One last question. Some pesticides directly enter in our body when we come in contact with them. Please throw a light on this.
Yes, definitely. There are a number of, I said, they are organic pesticides, especially endosulfan and other things. They are definitely penetrate into our, what you can say, through inhalation they go, through skin they can enter when we come in contact with them. So definitely they will go. That's what the people say, avoid those things.
Okay, sir. There is two more interesting questions from Dr. Shobhana. Can you say some of the assays in animals to test the neurological disorders due to xenobiotics?
Xenobiotics.
Okay, so there are animal models, especially monosodium glutamate. I said, especially in they leads to Parkinson's. Especially what this particular drug does. This is excitatory neurotransmitter. Okay, and along with that, it has a acetylcholine esterase inhibitor. So, acetylcholine, what you can say, increase the amount of acetylcholine by enhancing the enzyme called acetylcholine esterase. Okay, so because of that, it leads to a Alzheimer's disease. Okay, that's one of the few models what you can check. And there are some chemicals, Huntington's disease, we say that particular chemical which induces neurological disorders in the animals. I think some more animal model, I think Vijay Kumar will highlight more about it.
Thank you, sir. Now I would like to invite my friend Shiv Shankar to deliver the vote of thanks.
Then I'll stop sharing.
Yes, sir.
Sir, thank you very much, sir, for wonderful information and useful talk.
Excuse me, sir. I have a very small question from my side.
Yeah, sir.
As you told, eating the materials like sugar and maida and adulterated foods does harmful for the health, but the toxicity of the human body will recover it or not?
See, body has a very complex mechanisms. Okay, even you can give toxic. Even mythology also we studied, like a snake venom also people used to give in a small concentration so that the particular king will not be affected with the poisons. Okay, similarly our human body has all equipped thing to withstand these toxicants, but anything is overdose, definitely will affect. That was the sense.
ಅದೇ ಸರ್ ತಿಂದವ್ರೇನ್ ಬದುಕಿಲ್ವಾ ಅಂತ ಕೆಲವರು
that's what I wanted to say. From long time we are eating these things. Even though chapati eating people are there, people are avoiding. We should not eat chapati also, because it is made up of a wheat grain, which is contain a gluten, which is accumulated in tissues. See, everybody, humans doesn't have a same mechanism. Okay, some people can withstand it, some people can show that. So many people are affecting with the cancer nowadays. Maybe earlier people are not telling to other people, I'm affected with the death of the particular person is due to a cancer or not. Nowadays people are emerging and knowing about impact of cancer on everybody. Most of the people are dying because of a cancer. So one of the reason, I'm not talking about this is the reason, one of the reason is consumption of it. It's better to avoid it. At least we can save our next generation, avoiding our children's to the bakery item.
Spurti, you have any questions? Any important question? Then let us.
No, sir. Everything is fine.
Yeah.
So we
Thank
will finish with Shiv Shankar delivering the vote of thanks.
Okay, Shiv Shankar.
Yes, sir.
Yeah.
Hello, good morning, everyone. Myself, Shiv Shankar S., final year CBZ students of National College, Baswangudi, Bangalore. Firstly, my heartfelt gratitude to one and all who have gathered here for this wonderful webinar. It gives me an immense pleasure to deliver the vote of thanks for Dr. Veeresh P. Veerapur sir for conducting the webinar session on the impact of toxicants on human health. As sir said, all substances are toxicants. There is none that is not poison. But every substance has a toxic nature, but the right dose differentiates a poison and a remedy. Thank you, Dr. Veeresh sir, for enlightening us about the knowledge you have on adverse use of toxicants that impacts on human health, which further destroys his living. So that it is a great matter of pride for me that I have got a chance to thank this dignitariate person for his knowledgeable webinar. I lastly conclude my talk by saying this: health is like a money. We never have a true idea of its value until we lose it. So have a great care on it. I lastly thank all the participants for being a great listeners. Thank you all. Thank you, sir.
Thank you, Shiv Shankar. Now I would request Ramesh sir to welcome Ravikant sir for his next session.
Thank you very much, sir.
Yeah, thank you, sir. Thank you very much for your wonderful talk on behalf of the college, department, and all the participants.
Thank you, sir. Thank you very much.
Yeah. Very good morning. It is again my privilege to invite the other speaker of this webinar. I'm very happy to see the participants number, and from morning I think we have logged in at 7:30. I think US people, they have entered into that time and stay up to I think 10:45. I think we would have started the next session, they would have continued further, but we have given a little break. So after that, we are commencing this session. Now I invite Dr. G. Ravikant. Dr. G. Ravikant, a senior fellow, associate professor, Suri Siegel Center for Biodiversity and Conservation, Ashok Trust for Research on Ecology and Environment, commonly called A-Tree, Jakkur, Bangalore, Karnataka, India. Academic qualification: PhD Agri in Crop Physiology in 2004, US, Bangalore. MSc Agri in Crop Physiology in 1999, US, Bangalore. BSc Forestry in 1996 from Forestry College, Ponampet, South Kodagu, and is having the following research areas. Dr. Ravikant's research over last 20 years has focused on applying molecular tools in the context of tropical forest conservation and species recovery. He has led multiple research projects focusing on forest genetic resources that provide critical societal value. His empirical work has largely focused on conservation genetics and molecular ecology examining local pattern of spatial genetic structure across a range of economically important species. A core research focus has been on enhancing the conservation and sustainable use of forest genetic resources to deliver sustainable development. He has also focused on developing tools and research outputs to manage forest genetic resources of Western Ghats for forest landscape restoration and livelihoods. Dr. Ravikant's recent research has been on developing DNA barcodes for number of medicinal plants. He has used DNA barcoding to assess the extent of adulteration in herbal trade. He is also assessing the impact of harvesting on the genetic structure of plant populations. Ravikant's research interest can be broadly divided into understanding the genetic consequences of harvesting or habitat degradation or fragmentation, evolution and molecular systematics, biogeography and phylogeography, chemical ecology and bioprospective. His research integrates molecular ecological phylogenetics and niche modeling tools to address some fundamental research questions. His research group works across various taxa, including plants, amphibians, molluscans. He is having varieties of experience like senior fellow, fellow 2, fellow 1, and postdoctoral fellow and senior research fellow. He worked in this field or in this position. It is my privilege for all of us to have such a great, wonderful, versatile personality among us. We all are happy to know about your research work, knowledge, and success stories. Thank you for sparing out time for all of us for your busy and tight schedule to share your ideas with us. I welcome you to this international webinar, Dr. Ravikant Ji, to talk on animal, plant, environment, and human health. Need for One Health program. I welcome you, sir. Please.
Yeah, thank you very much, sir. Thank you, Dr. Ramesh, Professor Ramesh, for having inviting me and also giving this opportunity to address a large gathering. I would also like to thank rest of the speakers for covering most of my topic, especially Dr. Viresh, who actually started off the discussion with respect to plant products. I will be continuing further on that. And I now request Spurti, kindly, can you allow me to share my...
You should allow him to share.
Yes, sir. Just a second.
Yeah.
It's been a wonderful day from the beginning, and we have had wonderful talks. My talk is largely a general talk, where I will be actually showing the importance of the environmental health and how it is related to the human health. Okay, can you all see my screen?
Yeah, it is visible.
Okay, yeah, thank you. So I'll just
yeah.
One second. One just okay.
Yeah, it is visible.
Yes, thank you. Okay, so my talk is largely focused on the interactions we have with the animals, the plant, and the environment as a whole, and how the human health is influenced by the animal and plant health. And unless we have something called as the One Health program, it's very difficult to avoid future pandemics. Okay, so I will start off my talk with this slide, where I talk about the interdependence of the ecosystem. So what we have seen in the last century has been that humans have largely impacted the ecosystem, and the large scale human activities have actually destroyed the forest and led to habitat destruction of many organisms. Our industrialization as well as our developmental activities have led to increased toxicity, increased pollution in the environment. And this large-scale human activities has also led to ecological disequilibrium and loss of biodiversity. Both these in turn, that is the loss of biological diversity and increased toxicity and increased pollution in the environment, has led to increased risk of diseases. And loss of biodiversity has also led to a lot of wildlife coming into human habitations, and that has led to increased human wildlife interactions. And these two factors, increased human wildlife interactions as well as increased toxicity in the environment, have led to a lot of recent pandemics. So if you actually look at the pandemics, uh, you know you will see that there has been an increase in the pandemics over the last few years. And most of the microbes that are there are actually part of the wildlife diversity and often drive the population dynamics, whether it is wildlife dynamics or any other dynamics, or the plant population dynamics. For example, the density as well as the number of individuals in a population is actually determined by the microbial diversity, or microbes that actually control and manage the wildlife populations. But increasing anthropogenic environmental changes, that is, habitat destruction and loss of forest cover, has led to large scale land degradation, and this has led to altered microbial communities. And this habitat loss also has, as I mentioned, has led to increased interaction among animals and species, animals and humans. And there has been, because of this, there has been a spillover of zoonotic diseases. I'll come to what zoonotic diseases are, to the humans. And also, because of land degradation, the microbe communities have altered, and the microbe spillovers has actually occurred from humans, plants, as well, from animals to humans, as well as from plants to humans. So if you look at the pandemics over time, you will see that over time, there has been more or less occurrence of pandemics. But what you will see is, in the last 100 years, that is in the last century alone, the number of pandemics have increased enormously. In fact, if you look at the last 2 decades, the pandemics are actually much, much more than what we have also seen in the last century. So what we would be expecting is that there would be much more, pandemics that will be coming in the next few years. Okay, so we'll have to be prepared of that. And why does this happen? Because we should know that we are interconnected to the environment. We are interconnected with the animals, we are interconnected with the plants, and human health is interconnected with the rest of us, with all of us, with the entire environment. And I'll just give you a few examples of how pandemics, or how microbes have actually infected or impacted humans, that have actually come from, that have origin in the animal system. Just to start off the talk. So for example, to begin with that, I'll just like to introduce this term, but most of you are aware of it, but those who are new to this, the zoonosis is a term that is actually given to those diseases that come from animals to humans. Okay, so zoonotic. So if you look at it in the last few years, what we will see is that 60% of the existing human infectious diseases are zoonotic in nature. That is, 60% of all the infectious diseases have actually come from animal systems to humans, and at least 75% of the emerging infections or infectious diseases are actually coming from the animals. And 5 new human diseases appear every year. Out of every 5 new diseases that appear every year, 3 are of animal origin. And these human diseases, 80% of them also could be used as a potential bioterrorist, as a bioterrorist weapon. And therefore, we'll have to be very, very careful with regard to these pests and diseases. And first, for example, few examples that I will give about how, of the transmission of many of these viruses or many of the microbes from animal systems to humans. One of the foremost or well-known example is AIDS virus, or the immunodeficiency virus, which actually HIV virus, which actually came from the simians or the monkeys or the apes. And the human HIV is actually closely related to the simian immunodeficiency virus, that is the SIV. And we know that that most of the viruses from HIV viruses, actually, though there are multiple mutants of that, have actually come from the simians or the monkeys. And this is one of the classic example of how close interaction with animal systems has actually led to the virus entering into the human system. And this is, HIV virus has been a very, very important and devastating virus that has actually killed many individuals, especially in the African continent. And we also heard about this SARS virus, which we know has probably originated from the bats and the pangolins. And though we don't know about the intermediate host, but not very sure, but it is very likely that the ancestral virus, which was there in the bats and the pangolins, has actually got transferred to the minks, and from the minks it has actually slightly mutated and impacted the humans. And largely it has impacted the farm workers and those who work the farms, especially those who are in the meat industry. And Dr. Chandru also mentioned about how pigs and humans with their close interaction are very susceptible to such kind of diseases. So this is one example where, because of the close interaction, this particular virus has impacted the humans. And this was quite worrying virus, and because this is also a part of the coronavirus family, and this had actually impacted a large number of people again. Similarly, back home, very close to a place, is the Kaizenor forest disease, which again seems to have come from the tick bites, and from those animals, especially the monkeys that had this virus, which were monkeys and other small mammals are common hosts for this virus. And this disease again entered the human either through the ticks or through interaction with the infected animals or with the dead animals. And this again is there even now, but though it is very localized to places in Shimoga and that area, Sagar area, but nevertheless, this is an important forest disease that actually came from the monkeys to humans. And more closer home, or more importantly, the rabies is another important virus, which again is can impact anyone anywhere, because we have the dog population all over the country. And what we have seen is 99% of the virus, or the rabies virus, seems to have caused by the dog bites. And apart from dogs, this rabies virus has a number of other hosts, a number of other mammals, as well as especially a number of other mammals, especially mongoose, the foxes, as well as the bats. And what we have also seen that about one death occurs every 15 minutes because of this rabies infection, though it is not much talked about. But this again, nevertheless, is an important disease that has actually can come from animal system to the human system. Fortunately, there has been no report of human to human transmission, or not much is known about, or not much is seen of human to human transmission of this particular disease. So what we have seen is that from animal systems, human health is can be compromised if we have very close interaction with the animal systems, and if wildlife starts moving into urban areas or to areas where humans inhabit. If we start destroying the forest, we have already seen that there has been a lot of human wildlife conflicts, and there is more interactions of wildlife with humans. Nevertheless, there's also an important thing is that it's not just the animal health, the plant health is also important, and plant can also cause important diseases. And I'll tell you why plants are also important, because plants are an important source of our food, nutrition, and medicine. And in the recent years, what we have seen is that there has been a lot of influx of genetically modified crops, both of which, the source of food as well as genetic modified crops, can impact the food safety and security. And not only that, what we have seen also is that plants can act as there are pathogens that can cross kingdom and impact humans, from plant pathogens that can infect human health. Not only that, plants can actually also harbor pathogens which could be a pathogen for humans, so, and can cause indirectly plagues. I'll give you a few examples of this to each one of them, so that we understand this better, especially this section where where plants can cause severe damage to human and animal health. So first, coming to the plants as source of food and nutrition, and how the food is, or the plants are important for our health, because they directly affect our food security and safety. So as you know, plants are our major source of food and nutrition and medicine, and can directly or indirectly affect not only human health but also animal health. So when I talk about food safety and security, there are two aspects to it. When I talk about food safety, it is the absence of harmful elements in the food. That is, harmful elements could be any kind of microbe. So absence of any kind of harmful elements could not just be microbes, but also other toxic chemicals, including heavy metals. Though I will not at this point of time talk about heavy metals, but I would talk about the microbes that are there in the plants. But not only that, food security. When I talk about food security, it is the ability to get consistently safe and nutritious food. So food safety and food security are important aspects of human health. So when you come to food security, there are the world population is actually facing two extremes, and India is actually in the top of both. We are actually having both the problems. At one end we have malnutrition, on the other end we have obesity. So malnutrition is the lack of nutritious food to sustain life, and obesity is increased weight. And so increase in obesity rates has been seen in the adults and children, especially in this particular century, and this is also termed as an epidemic. And as we know, India also is an capital of diabetes, and this diabetes is again due to higher increased levels of obesity. And if you look at malnutrition, India is one of the top countries which is having high prevalence of malnutrition. In fact, in year 2000, about 7 lakh children under the age of 5 died due to malnutrition, and only about 6.4% of children are known to get proper diet between the age of 0 and 2 years. That is, balanced diet is available only to 6.4% of the people. Okay, and if you look at overweight and underweight in India, India is alarmingly high, has high number of people both underweight and overweight. So as I told, India has both the problems of obesity as well as malnutrition. And these are the statistics to show that Indian men are overweight. About 9.8 million 8 million people or the Indian men are overweight, and about 20 million women are obese, and that is about 3.7 to 5.3% of the global population. And we also have high number of people who are underweight. So 40% of the global underweight population is in India. So these two again are important things that we need to consider when we're talking about the food safety or security. So, and if you look at the Indian diet, the calorific intake of is deficit of vegetables, fruits, and legumes, as well as animal proteins compared to a reference diet. So, if you look at the country, Karnataka is in a way, slightly better with respect to legumes and animal source proteins. But if you look at vegetable and fruits, you will see that there has been a severe calorific deficit with respect to intake of these diets. The darker color here represents a high calorific difference, whereas the light color actual difference between actual consumption and reference diet. So if you look at legumes, Karnataka is quite good with respect to legume consumption or protein consumption through legumes. But whereas with respect to vegetables and fruits, we are actually lagging far behind. And with respect to food safety, we'll find that worldwide 1.5 million people died due to contaminated food and water. And contamination of fruits and vegetables is largely by these two microbes, Salmonella and E. coli, which can cause significant burden on the healthcare systems every summer. In fact, in India alone, if you take about 10 lakh cases of acute diarrheal diseases are reported, and then 3.6 lakh deaths occur every year, largely due to the foodborne contamination. And so foodborne outbreaks are important source of illnesses, and these foodborne illnesses are largely due to contaminated fruits and vegetables, which are the major cause of foodborne illnesses. And in fact, what we have seen is 20% of foodborne outbreaks are by E. coli, and we have also seen that other pathogens like Shigella and Campylobacter also has been observed as important pathogens, and these are largely due to contamination or irrigation of sewage water, and this sewage water coming either from lakes and ponds, and that actually has been used to grow vegetables. And if the vegetables and greens especially are not cleaned and used, there is a possibility of food outbreak and contamination of or food poisoning that can occur due to these foodborne diseases. And what we have seen is also that half of the known outbreaks are caused by multiple strains of Salmonella, and this is actually linked to largely due to tomatoes and cucumbers or watermelon, and melons. That is because we largely eat these as fresh fruits and vegetables. The cucumber, for example, is eaten fresh, and therefore the possibility of contamination through them is much higher. So unless we clean or wash these vegetables in clean water and wash repeatedly, there is a high possibility of contamination. And there have been cases and studies to show that surface of fruits has been a favorable environment for many of these microbes to thrive and survive. And therefore, there is a possibility that they can infect humans. In fact, the betel leaf, the pan leaf that we all consume, there has been a nationwide study, and they have shown that 6% of the pan leaf had contamination of salmonella. In fact, if they had, if people don't wash or if they don't use lime on the on the betel pan, there's a possibility of these contaminants actually entering the human body. So that is with respect to salmonella contamination, and these are studies to show that salmonella can actually not only be on the surface, but in recent years, what people have shown is that the salmonella has actually entered the inside of the fruits and and vegetables such as cucumbers. So these are the different strains here of cucumbers, and what they have shown is that they are not only in the surface, but even inside there seems to be contamination of Salmonella enterica. And this is a study that was published last year, where they have actually shown that sometimes washing also doesn't help. So unless these fruits, these vegetables are grown in a environment which is free from contamination of pathogens, free and irrigated it with good clean water, there is a possibility that these microbes can enter inside the fruits and vegetables. So that is an important study that has been shown last year. What is also important, so that covers the topic on food safety and security, but I want to talk more about the other aspect, that is plants also susceptible to pathogens and can harbor human pathogens and can directly or indirectly cause plague. And so if you look at one of the greatest famine, in the potato famine or the Great Hunger, the Great Famine, this was, which occurred in the 18th century, and which led to mass starvation and disease in Ireland. In fact, so much so that 20 to 25% of the Ireland population was wiped out. Okay, so about it, and it killed about 1 million people during that time, during the 18th century. About 25% of the entire population died because of this particular potato famine, which actually impacted or killed large populations or the large areas of potato cultivation, and this led to starvation and death of so many people. It was also called, the 1947 is actually 1847 is also called as the Black 47, which impacted impacted very severely and wiped away 20 to 25% of the population. And if you look at other plagues, for example, if you look at the Nipah virus, we will see that Nipah virus actually seems to have come from the bats through the plants. So how has it come? Though a number of pathogens shared between plants and humans are less compared to pathogens shared between animals and humans, many times plants indirectly play a role in the spread of pathogens harbored by animals. And this Nipah virus is one such example. So if you look at how Nipah virus came, it is assumed, or it is now proved, that Nipah virus actually came from the bats. And how did it come to humans? This was largely because the bats, fruit bats especially, they fed on the date palm sap, and people who collected the date palm sap, which was infected with the Nipah virus, and consumed this date palm sap, they got infected with the Nipah virus. And the bats also were responsible for transmission of this Nipah virus to pigs, and because people also consumed pigs, this particular virus entered through. So there were two routes: one directly by consumption of date palm sap, and the other through the pigs. And this came to humans, and from humans, it actually spread from human to human, and there was man-to-man transmission of this particular virus. So here again, you will see that the plants, animal, and human, so all are related. The bats fed on the fruits, and the fruits were fed either by the humans or by the pigs, which actually got infected, and that this led to the Nipah virus transmission throughout the globe. Similarly, if you look at other plagues, so this is again an important bamboo plague that most of us are aware of, and many people in Malnad would know that mass flowering of bamboo is often feared, because and it's not just in Malnad region but in the northeast region, which occurs every 40 to 50 years, where a large scale flowering and fruiting of bamboo occurs. And when this occurs, there is a large scale availability of bamboo seeds in the forest, and because of plenty of food available, the rat population blooms because there is a food available. And once this food is over, and then you have a large number of rats available in the forest, they start moving from and because of non-availability of continuous supply of bamboo seeds, they start moving from the forest into the human settlements. And there have been reported that the plague or the pneumonic plague can cause because of the rat flood in the human settlements. And this occurs as the rats multiply in response to the temporary windfall of bamboo seeds, and they leave the forest to forage for stored grains as soon as the bamboo seeds are exhausted, which in turn causes devastating famine. And a lot of people are aware of this. Okay, this I covered. So a crop pandemic would be as devastating for biodiversity as COVID-19. This threat is far from being theoretical. Already we know that dozens of varieties of native and wild species are in danger of disappearing due to temperature changes, or largely because of climate change and loss of habitat. And so these new diseases seem to be not only impacting humans, but also new diseases are also endangering our crop plants, and especially those with respect to wheat, potato, and bananas, and other wide relatives of crop plants. And there's a possibility that these can impact humans. So I will talk about now give examples of cross-kingdom pathogens from plants to actually which have impacted humans. So the major questions here is, do pathogens merely exist in plants, or do they have a wider host? And is there a possibility of these pathogens to jump from plants to animals? If you look at the plant cell and animal cell, plants and animals have a very different structure. The plant cell is characterized by a plant cell wall, which is actually very hardy material, and which actually acts as a significant barrier for pathogens to enter. Whereas the animal cell doesn't have that much of a physical barrier, and therefore animal cell is much more compatible for for the microbes to actually enter. But if you look at the mechanism of the pathogen which involves adaptation, colonization, virulence, they are more or less common across the host. And once the pathogens overcome the host defense by either disrupting the cell wall or by producing virulence factors such as toxins and enzymes, they can actually cause diseases. And the virulence factors trigger the expression or repression of host genes. And once these pathogens are able to overcome these virulence factors, they can cause diseases. So I'll give example of cross-kingdom transfer. This was actually again a recent study that was actually conducted in Bangalore, with a number of institutes, including the Indian Institute of Science and University of Agricultural Sciences, which was published about 2 years ago, where they showed that this particular salmonella can from a plant can go into the root from the if the infected plant is eaten by the rats. Okay, and this rats if they this salmonella can infect the rats, and if the infected rats the fecal matter from the infected rats can enter the plant again. So there is a cross kingdom transfer of salmonella from the rodent feces into the soil, and from the soil to the plants again. So this is a circle that they have actually shown, and they have shown that both in the there seems to be infection of the rats if they eat the salmonella infected plant, and from the from the fecal matter the plant can get infected again. So this is a wonderful study that they showed that there is cross kingdom transfers. I'll just give you a few examples of plant fungal infections that can cause infection in humans. This just to let you know that you don't have to be panicked because these are only seen in immunocompromised individuals. especially those who have had surgeries, or those who have actually taken cancer treatment. For example, if they have taken chemotherapy and where their immune system is actually very low, during those cases, these infections have been seen. For example, this particular fungi called Alternaria infectoria, which actually causes blossom blight in plants, seems to cause eye diseases, contact lens related infections, and this again was seen only in the immunocompromised individuals. So this is one example of a fungal being infected, but there are a number of other fungus that seem to have actually infected humans, and all these again are infected humans because of either surgical site infection, or because of blood infection, or because of eye infection, largely because people, sometimes those who are using contact lenses keep them in different places where these microbes, these fungus occur, and if they don't wash and keep it, store it, put the contact lenses back, there's a possibility that these plant fungal infections can cause human infection. And this has been actually reported over the years. If you look, I've given some references at the bottom. So if you look at these papers in the last 15 years, a number of these fungal infections have actually caused infections in humans. Similarly, there are a number of bacteria that have actually also caused human infections in humans, especially this particular Rhizobium radiobacter, which is a very, very common bacteria, which can infect large number of eudicots, which can infect large number of plant systems and cause crown galls, has actually been reported to cause urinary tract infection, as well as catheter related bacteremia, and this can actually cause death. Fortunately, again, these don't transmit from human to human, but from plant to humans, they have been seen to infect. So similarly, there have been other bacterial infections that have actually caused infections in humans. These plant
Dear all, can you please mute your microphones?
You can go ahead, sir.
Yeah. So these plant bacteria, which actually cause disease in plants, especially in maize, onion, rice, tomato, and other fruit-bearing plants, seem to have actually caused infections in human systems. So you see that not only fungus and bacteria, but there are also some viruses that have actually been reported. And this is the tobacco maize virus, which is all of you would have studied. This has actually can cause, in many of the tobacco and tomato Solanaceae members, which actually it causes stunted growth. They have seen actually that this particular virus can infect the lungs of humans, and this actually, which actually is especially seen in tobacco smokers, and especially not only smokers but those who actually consume tobacco products, and they have seen that this TMV virus can infect, and it actually has been found in the lungs of both active and passive smokers, as well as patients who actually consume or chew tobacco, and this can actually cause severe problems in the lung. So again, the viral from the plant seems to have infected humans. And there are other eumycetes, which are other again plants, which again have seemed to have come from the animal system, sorry, plant system to the animal system. And this was actually first found in a patient who was injured in the Afghanistan war. And here, when he laid down, when he was infected, he was he was injured in the war, and this particular person was infected by this particular plant oomycetes, which was like one of the first report, and this was again reported in this particular decade. And so if you look at these phytopathogens, whom do they infect? With few exceptions, most of these plant pathogens have largely infected immunocompromised or post-surgical patients. And in the last few years, we have seen that these are actually infecting people who have undergone chemotherapy and whose immune system has completely gone down. And if they, in fact, in the last few years, chemotherapy patients are actually advised not to consume fruits and vegetables in raw form, because they can get infected by plants, by plants, and because their immune system is very, very weak. However, active immune disruption of Pseudomonas syringae has actually been also seen, and these are opportunistic pathogens. And we have, in the recent years, we've also seen that they can also infect healthy, small percentage of healthy populations or healthy human populations as well. And this we can actually see, in the last few years, all of you, last few days, all of you have also heard about black fungus. And this black fungus, or mucor mold, it actually is a common fungus that is actually found in the soil, plants, and as well as in manure. Okay, so this again, actually has started infecting humans only in the immunocompromised individuals again because of COVID-19. It's actually not. It's not going to affect any of the healthy humans because we can actually deal with it because we have a good immune system. But if your immune system is compromised again because of, for example, COVID-19, we have seen that this particular fungus has actually started infecting so many people. In fact, this is the first time that people are reporting so many people infected with this black fungus. Again, this particular fungus is seen in soil and plants and manure, and this has actually come. So this is a very good example that has actually come in recent days. So after infecting humans, can humans multiply this particular virus or this bacteria or the fungus and spread? Fortunately, till now, we don't have any information or any case where the fungus or the bacteria or the virus that has actually come through the plants to the mammalian cells, and from mammals it has actually spread to other mammal systems. Whereas, however, there have been some studies in the labs where they have actually shown that, especially with respect to Providence virus, where it has actually shown that this particular virus can not only infect the plant, but from plant it can enter the vertebrate cells as well as in the insect host. So and you know that viruses are really very, very quickly they can adapt, and replication of plant viruses in animal cells is possible, and raising the possibility that these viruses may be able to cause human infections. And because these viruses have high mutation rate, there is every possibility that plant viruses in some day in the future can gain upper hand in the evolutionary arms race and can infect humans and can cause severe diseases like the coronavirus. So we have to be very careful that these viruses can, and if they get the right mutation, they can actually cause human diseases. So, and the other reason why we should also consider plants is in recent years, we're also looking at, we're also seeing that, large number of food products or crops are getting genetically modified. And this has actually has a very, very serious issue with respect to our food security and food safety. And three major health risks have actually been reported with respect to genetically modified crops or genetically modified foods. One is the toxicity, second is allergic reactions that many people have reported, and the genetic hazards. That is, there is a possibility of gene transfer from the human gut bacteria through the horizontal gene transfer, and this is actually have to be very, very carefully considered. So food from the plants, or plant from microbes from the plant, have actually also have equal opportunity or equal possibility of entering human systems. And therefore, in the last few years, in the last last decade, there has been a large emphasis on something called as the One Health framework. So One Health framework involves people who protect human, animal, and environmental health, coming together to coordinate, communicate, and collaborate to achieve the best health outcomes for people, animals, and planets. To just to give to give background about One Health origin, this actually, the One Health concept was created in 2004, where the novelty was there is incorporation of ecosystem health, including that of wild fauna. And a large number of European ministers actually met, and the World Health Organization met on June 15th, 2017, and paved the way for this ambitious integrative approach called as the One Health framework, which actually recognizes that human, animal, and environment health cannot be separated, and they are all together. And therefore, when you're talking about diseases and pandemics, we have to also look at the entire total, the total health of the entire environment, the health of the entire animal system, as well as plant, when we are actually talking, because the health of the animal, the health of the plant, and the environment is important, because these three can impact the human health. So this, the concept includes, issues such as zoonotic pathogens. It actually started off with zoonotic diseases, where it was actually dealt with the niche adaptation, epidemiology, vector transmission, and food safety and food security, biosecurity. But in the later years, in the last few years, they've also included a large number of issues in the One Health concept. So now the One Health concepts include not only zoonotic diseases, but also looks at antibiotic resistance, food safety and security, the vector-borne diseases, the environmental health, mental health, occupational health, as well as the plant health, and much, much more. So all these are now actually given wider coverage. And so now the World One Health team actually consists of people, the doctors from the animal systems, the human health doctors, the animal doctors, the plant doctors as well, and are discussing that emerging of these diseases. So I would like to end my talk with this particular slide. When we know in the recent years, and in the last 2 years, the government of India also has been looking at this overlapping zones, and actually is launching a mission called as National Mission on Biodiversity and Human Health. And this has been approved by the Prime Minister's Science and Technology and Innovation Council, though because of the pandemic and the lack of funds, this was actually not kicked off. But in the next 2 or 3 years, this particular mission is going to be kicked off, which looks at biodiversity and human health in a holistic manner. So where this particular mission actually has about 8 different components, from starting from cataloging and mapping life of India, looking at how biodiversity impacts our ecosystem services, how biodiversity and climate change and disaster risk management impacts humans, looking at biodiversity and agriculture component, which I am part of. And the other important aspect of this mission is looking at One Health and zoonosis, as well as medicinal plant and human health. So these two components, especially looking at which One Health and zoonosis is an important component of this program, as well as looking at biodiversity and bioeconomy. And also this program actually looks at how biodiversity can be used, and how people can be made aware of this biodiversity by building capacity building programs and outreach activities. So this particular mission aims to strengthen the science of restoring and conserving India's natural heritage and embed biodiversity as a key consideration in all the developmental programs. So this mission is going to be launched soon. And so in conclusion, I would want to say that animal and plant infections may have critical impacts on human health and safety. And people have also covered about how environmental pollution can also critically impact human health. So One Health program is the need of the hour, where we actually not only have to look at the animal health and plant infection, but also look at the ecosystem health as well. And recent genomic data reveal cases of horizontal gene transfer, not only between bacteria and the host plant genome, but also between bacterium and other eukaryotes. And therefore, we have to be very, very concerned about this. So understanding the disease processes and outcomes across kingdoms would help us to stay prepared to contain any outbreak. And this can be achieved by building network of understanding and trust that is much needed in times of outbreak and other crises, such as what we are facing right now. Thank you. And if there are any questions, I will take it.
Thank you, sir. I'll be taking few important questions. So there's this question which says, is there any role of microfinance in human body that can give resistance against pathogens?
Microfinance, is it? I didn't understand the
question.
Yeah. Microfinance.
Yeah, that's what they mentioned, sir.
Oh, okay. I am not aware of that. Microfinance, I don't know. Or microbes they are talking about.
Yeah, might be.
Microbes, it may be microbes.
Yeah, microbes. Yeah, so human body is naturally resistant to...
1 minute.
Yeah.
Am I audible? Ma'am,
Yeah.
1 minute. We are asking a few questions. Later on that, we'll let you...
Madam, you can ask later, madam, at the end. After finishing these questions, you can ask.
Yeah.
Requesting everyone not to interrupt.
Yeah, sir. Microfauna means whatever
Okay, microfauna. Sorry, I heard it as microfinance.
Microfauna that is present already in our body.
Okay. Yes. Okay, yeah.
I mean, adapt to itself inside.
Yes, that's a very good question. Actually, microfauna are okay. That's a good question. So actually, microbiome in recent years has been a wonderful study area in the recent years. Not only human microbiome. The human microbiome consists of a lot of microbes that are supposed to be. We have both the good microbiome as well as the pathogenic microbiome. The human microbiome or the good microbiome that we have in our body is able to resist most of the pathogens. In fact, if just to give an example, if you people, many people know of this example, that if you are suffering from acidity, people generally consume curds. Okay, so curd also has a good microbiome that actually helps in combating many of the disease-causing or bad bacteria or the bad microbiome that is there in the body as well. So human microbiome is an interesting area of study that has actually, in the last few years has been given a lot of importance, and people are actually studying. I mean, actually, if I want to, I mean, it's a very big area, and I can go on talking about it, but maybe we can talk personally, I mean, separately on that issue. Thank you.
Yes, sir. Next question is there any chances of using these pathogens as bioweapons?
Yes, in fact, now people are very aware that these can be used. In fact, we are still awaiting research into the origins of COVID-19, and people are wondering, there are some hypotheses saying that COVID-19 actually was being developed as a bioweapon. And so not only COVID-19, but there are a number of pathogens that people have actually cultured and cultivated, and not only in China, but in many parts of the world, that people are actually actually having these potential microbes that can actually be used as a bioterrorist or bioterrorism, can cause can cause epidemics in different parts of the world. So there is a possibility.
Next question is there possibilities of plant pathogens which affect pollinators like honeybees? And if there's example that you can mention?
Plant pathogens impacting honeybees is not yet known. I mean, we don't have any evidence right now, but there have been pathogens which can, impact directly, honeybees. In fact, one of the reasons of honeybee decline is also because of not only pollution, but also because of a fungal infection that actually has wiped out many bee population. So that is also important. But from plant directly impacting honeybees is still now not known.
Sir, last question. We have very few antifungal drugs. Is this a pressure or threat on the pharmaceutical industry to bring more cross-infection drugs to maintain human health?
Yes, in fact, that's also a good question. And in recent years, there has also been a lot of research regarding that. And in the years to come, you will actually find a lot of antifungal, antiviral drugs that are going to come into the market. In fact, all over the world now, people have actually started looking at antifungal drugs, and a lot of research is actually happening right now. Yeah.
So there's one last question which I found interesting. Is there any identified gene that can convert pathogen to infect any of the organism? If yes, then by silencing it, can we control entrance of pathogens which comes from the wild?
Okay, that's a good question, but there is not just one gene. In fact, virus, for example, has a number of hypervariable regions, and because of this hypervariable region, you cannot actually, it's not one gene. So, and there are a number of hypervariable regions in the virus, and therefore they can mutate, and therefore new and new strains can keep coming, and you can't mutate that particular part. You can't silence that, because if you silence one, there's another one coming up. So there are a number of regions like that. In fact, the reason the viruses are so successful in infecting people is because of the hypervariable regions, and they can actually mutate faster than any other organism and can cause diseases. Yeah, thank you.
Thank you, sir. Now I would like to invite my friend Srishti Vijay Kumar to deliver the note of thanks to this session.
Yes, Srishti?
Yes, sir.
Yeah, go ahead.
Thank you, Spurti. Namaste to one and all. First of all, I would like to thank the principal madam and Ramesh sir for giving me an opportunity to speak on this auspicious occasion. On the behalf of the entire institute, I would like to thank the Honorable Ravikant sir for sparing time to grace this webinar with the topic Animal, Plant, Environment and Health. We got a detailed explanation about the interaction between animal, plant, environment, and how it impacts on our health. I express my gratitude from bottom of my heart for the sophisticated explanation, which kept us spellbound. Thank you, sir.
I would also like to invite Rashmi to speak few words. Is Rashmi there?
Rashmi? Rashmi? Rashmi?
Hello. Yes, sir. Yes, sir. I'm
Yeah.
there.
Yes, yes.
Yes, sir. It was an honor to have been asked to speak now. This is an event, international webinar on current trends and impact of toxicants on biodiversity and human health, organized by the Department of Zoology, the National College, Baswangudi, Bangalore. On behalf of the National College, Baswangudi, and the entire fraternity of the Institute, a sincere and heartfelt thanks to Dr. G. Ravikant sir, who spent his time gracing the occasion on the topic Animal, Plant, Environment, and Human Health, a Need for One Health program. Today, we had an opportunity to listen to your thoughts, and this is definitely going to encourage us in our future events. I would like to express my gratitude for giving excellent coverage to the topic, sir. I would also like to extend my sincere thanks to all the teachers and the participants for joining the event and making it successful. Thank you.
Thank you.
Thank you, Rashmi. Now I would like to invite Harish sir to welcome our next webinar speaker, Vijay Kumar sir.
Now the next session. Thank you, Vikant sir.
Thank you, sir.
Thank you. Wonderful
Pleasure.
presentation. We enjoyed and the students are enjoyed all the.
Good afternoon to everyone.
Yeah.
It was a wonderful session.
Good afternoon, everyone.
So 5 days we are working from Monday till Friday.
It gives me a pleasure to welcome Dr. Vijay Kumar Yash, Associate Professor in Department of Pharmacology at Sri Siddhaganga College of Pharmacy, Tumkur. Dr. Vijay Kumar Yash, sir, also having a great career achievement, awarded second place in poster presentation in 2016, 21st Annual National Golden Jubilee Convention, Manipal University, and also awarded third place in poster presentation at ICHTR in 2015, Manipal University, and awarded second place in poster presentation at Annual Convention, Capticon. Articles published are 32, short-term training programs attended 12, sponsored research projects are 3. He also worked as a co-principal investigator in RGUH Research Grant 2017 fund. The principal investigator, RGUHS Research Grant 2018 fund, and sir is also having a great academic experience of 15 years. And now he is here to deliver a speech on toxicological research in animal model. Sir, we highly welcome you for your sharing of knowledge. Thank you.
Good afternoon, sir. Vijay Kumar sir. Yeah.
Thank you.
Welcome to our webinar.
Thank you, sir.
Please, you can start your presentation.
Umesh sir, can you please give your co-host permission to Vijay Kumar sir? Actually, I'm unable to do it because there are 3 of them as co-host.
Okay, you can give, madam. Spurti, you can give.
Sir, I'm asking you to give, sir, because there are 3 of them, so I'm not able to do it.
Right. Notice. Spurti, it is not possible for me.
Just a second, sir, 1 minute.
Yeah.
Requesting all to please mute your microphones.
Yeah. You got it?
Disregard it. Still it is disabled, sir.
No. She's enabling, sir.
Service on 30th April 2021. Further, as per the recommendations of the Department of Conservation, these services are being utilized for the completion of classes of first and fourth semesters. First and are being utilized for the completion of the were utilized for
The noise is coming. Yeah. Purti, what happened?
Sir, can you only play the PPT, sir?
No, it is not there with me. So you make him a co-host.
Sir, it is not possible, sir. There is some problem. I just called Vijay Raghavan, sir. Maybe the timing of the link is problem. So can you please get the PPT?
Yeah, no. What problem actually now is making co-host or link problem?
Co-host ಮಾಡಕ್ಕೆ.
Okay.
Yes, sir. Co-host ಮಾಡೋದಕ್ಕೆ problem actually. So ನೀವೇ PPT ನ ಇಸ್ಕೊಂಡು ಶೇರ್ ಮಾಡ್ಬೋದಾ ಅಂತ.
ಇಲ್ಲ ಅವ್ರು ಇಷ್ಟು ಮಾಡಿದ್ರೆ ಈಗ ನಿಮ್ಗೆ ಆಗ್ತಾ ಇಲ್ವಾ? ಅವ್ರಿಗೆಲ್ಲ ಮಾಡಿದ್ರೆ ಈಗ ಏನಾಗ್ತಾ ಇದೆ? ಏನ್ ಪ್ರಾಬ್ಲಮ್? ವಾಟ್ ಇಸ್ ದ ಪ್ರಾಬ್ಲಮ್? Busy, maybe is in the class. ಪ್ರಾಬ್ಲಮ್ ಇರುತ್ತೆ ಅದು.
ಇಂಟರ್ನೆಟ್ ಗೊತ್ತಿಲ್ಲ ಸರ್. ಮಾಡ್ಕೊಡ್ತೀನಿ.
ಅದಕ್ಕ ನೋಡಿ, ಈಗ ಅವರು ಲಾಸ್ಟ್ ಪ್ರೆಸೆಂಟೇಶನ್ ವೆಯ್ಟ್ ಮಾಡ್ತಾ ಇದಾರೆ. ಬೇಗ ದೇ ಆರ್ ವೇಟಿಂಗ್ ಫಾರ್ ಲಾಸ್ಟ್ 5 minutes ಇಂದ ವೇಟ್ ಮಾಡ್ತೀನಿ. ಸ್ಫೂರ್ತಿ, 1 minute.
Yes, sir.
1 minute. Where that sound is coming?
ಹಾಕಿ ಫೋನ್ ಮಾಡಲಿ ನಿಮ್ಗೆ ನಿಮ್ದಾನಂದ
ಸರ್. ಒಂದು ನಿಮಿಷ ಸರ್, ಬಂದ್ರು.
ಟ್ರೈ ಮಾಡಿ ಸರ್.
ಇಲ್ಲ ಮಾಡ್ತಾರೆ. ಆಯ್ತಾಯ್ತು, ಎಲ್ಲದಕ್ಕೂ ಆಯ್ತು. ನೀವು ಇದು ಈಗ ಒಂದು
NCB 18 ಸರ್. NCB
18. Which Raghavan? How it is? Yeah. Check you now, Spurti. Spurti.
Wait, sir, 1 minute, sir. I'm solving the issue.
Okay. Sir, Vijay Kumar sir, ಕೆಲಸ ಇದೆಯಾ?
Yes, sir.
Okay. ಒಂದು ನಿಮಿಷ ಆಯ್ತು.
Hello. Am I audible?
Yes, yes.
Yeah. Because some of the net problem or something is the problem there. Let me discuss about one of the main question I asked to Ravikant sir. That is, zoonosis actually from from wild animals, it is passed to human beings, isn't it?
Yes, yes. Yeah.
Yeah. Because many of the pathogens which show the host specificity. That means they infect only a specific host. How
yes.
they can, get this infection, broad spectrum infection to any of the animals. How can they infect?
Yeah, so in recent years, what we have seen is that many of these, infections, or sorry, microbes, seem to have, are getting mutations that is enabling them to actually adapt or jump from one kingdom to another. In fact, not just from one animal to another, but actually are jumping from one kingdom to another. In fact, that is what I showed about, from plant kingdom to animal kingdom. But it is more easier to actually jump from one mammal to another mammal species, because very few mutations are required. But it is largely seen in people who have very low immunity, and from that they start spreading from low immunity people to also people who are very healthy. So it is actually a small mutations that are actually occurring, and these mutations can occur because of number of factors, including what people have actually been commenting about, being pollutants being actually increasing in number or because of high number of pollutants in the atmosphere. So that's also because of that. That's one of the reasons.
Thank you, sir. Ramesh sir, he is the co-host now.
Yeah. Vijay Kumar sir. Yes. Vijay Kumar sir. Is our voice is audible? Hello.
ಸರ್ ಈಗ ನಾವು ವೀರೇಶ್ ಸರ್ ಇದರಿಂದಾನೇ ಮಾಡಿದೀವಿ. ಇಟ್ಸ್ ಓಕೆ.
ಯಾಕೆ ಆಯ್ತು. ಓಕೆ, ನೋ ಪ್ರಾಬ್ಲಮ್.
ಶೇರ್ ಮಾಡ್ತೇನೆ.
ಓಕೆ, ನೋ ಪ್ರಾಬ್ಲಮ್. ಯು ಕ್ಯಾನ್ ಸ್ಟಾರ್ಟ್ ಸರ್. ನಾವು ನಿಮ್ಗೂ ಕೋಸ್ಟ್ ಮಾಡಿದ್ರಿ ಈಗ.
ಮಾಡಿದ್ದೀರಾ? ಒಂದ್ ಸೆಕೆಂಡ್ ಚೆಕ್. ಅದು ಒಬ್ಬರಿಗೆ ಕೊಡ್ತಾರೆ.
ಅಲ್ಲ, ಇಷ್ಟೊತ್ತನ ಕೊಡ್ಗಿತ್ತಲ್ಲ.
Sir, now Veeresh sir and Vijay Kumar sir, both of them are co-hosts, sir. So they can share. Anyone can share.
That's what I'm asking.
Who is sharing now, sir?
I am sharing, Veeresh.
Okay,
just
okay,
give
sir.
to him.
Okay. Yes. Go ahead, sir. Yes. It is visible.
ಅಲ್ಲ ಸರ್, ಒಂದು ನಿಮಿಷ ದೊಡ್ಡದು ಮಾಡ್ಕೊಡಿ. ಇದನ್ನ ಹೇಳತ್ತೀನಿ ನಾನು ನಿಮಗೆ, ಇದು ಅಡ್ಡ ಆಗುತ್ತ ನಿಮಗ ಅಂತ.
ಇಲ್ಲ ಬಿಡಿ ಸರ್, ಅದೇನು ಅದು ಟಾಪಲ್ಲಿ ಇದ್ರೆ
ಇದನ್ನ ಮಾಡ್ಬೇಕು.
ಮಾಡ್ತೀನಿ.
ವಿಡಿಯೋ ಮಾಡಿ ನೋಡ್ರಿ.
Yes, sir. It is visible.
Okay, sir.
Yes.
Good afternoon, sir.
Good afternoon.
Good afternoon. Okay, sir. My topic is toxicological research in animal models. Since from morning all the speakers has given the information about some toxicants, let us move with the animal study. That means different animal models, what we are going to use in the toxicological research. The research involved the laboratory animals which are necessary to ensure and enhance the human and animal health and protection of environment. See, we are going to use the animals. Why they are important? In absence of human data, research with experimental animal is the most reliable means of detecting important toxic properties of chemical substances and for estimating risk to human and environmental health. The basic reason why we are using the animals in the research is the disease process in the human and animal have similarities, whereas in the cell system or in the cell system contain or manipulate only a part of the organ system, whereas the computer models are lack of the complexities of the living entity. The animal model is defined as a specific combination of animal species, challenge agent, and route of exposure that produces a disease process or pathological condition that in multiple important aspects corresponds to the human disease or condition of interest. In the sense, whenever we are selecting the animal model, we have to have the combination of the specific species of the animal and the inducer, that means the disease causing agent, and whether the disease causing agent is going to cause the disease in a particular pathway or not, or pathological condition, how it is going to produce. All these things are important for us. The animal models are the best analogs to human conditions, but no theory can be provided or refused by analogy. The truth is, the evidence in the animal can be a powerful device in supporting virtually theory. In the sense, these animal models are giving the evidence for the further disease-causing procedure or how we are going to cause the toxicity. See, we know that in the toxicology, we are going to use many different animals. Like here, it has given the information that mice, largely we are using the mice for toxicity study. Next, the rats, amphibians. We know that this mice, rat, guinea pig, even the fish, honeybee, including the earthworm, rabbits, etc., we are going to use. According to the WHO, nearly 2 million people die worldwide from accidental poisoning. In the sense, it is important the toxicants are going to cause a major death. See, the statistics of 2019 shows that there are 25 lakhs of the people are affected by these toxicants. Toxicology classically has been defined as a study of poisons concerned with the adverse effect of xenobiotics. And Casaret defined it as a science that defines the limit of safety of chemical agents for human and animal population. Toxicological screening is very important for development of new drug and for extension of therapeutic potential of existing molecules. Very importantly, whenever we are going to develop the new drug in this condition, that means before going to the human treatment or administration, we need to know its toxicity, and after even during the treatment also, what is the extension of its therapeutic potential, we need to understand, we need to study the toxicology. In the toxicology, need to prove drugs are safe before the administration of the human being, and toxicity tests are mostly used to examine specific areas or specific endpoints such as cancer, cardiotoxicity, skin or eye irritations, etc. This testing helps to calculate no observed adverse effect levels, and also to fix the dose. It is useful in particularly the clinical trials. To do the toxicology study in the animal, we need to follow the biomedical ethics. Before conducting any toxicological testing in the animal or collecting the tissue or cell line from the animals, the study should be approved by institutional animal ethics committee, or the protocol should satisfy the guidelines of local governing bodies. Let us see few governing bodies. Some regulations we have to follow, or the guidelines we have to follow when we are conducting the toxicological studies. In India, the Committee for the Purpose of Control and Supervision of Experiment on the Animal comes under the Ministry of Fisheries, Animal Husbandry and Dairying, Department of Animal Husbandry and Dairying, is we are going to follow the procedures or the protocols of this particular, that is CPCSEA. The CPCSEA has a goal of promote the human care of animals used in the research to provide specifications that will enhance the animal well-being and quality of the research with the objective of functioning of institutional animal ethics committee. The CPCSEA was formed in 1960. Later on, earlier it was in the Ministry of Agriculture. Later, it is under the recommendation of Animal Welfare Board of India. It is converted to CPCSEA. Now it is under the Ministry of Fishery, Animal Husbandry and Forestry. The Institutional Animal Ethical Committee is a local body. In each research center, they should have the IAEC. Its functions are to approve it is approved by the CPCSEA, and it has empowered to permit the experiment whenever we are going for the animal experimentation, whether it is toxicological or screening or whatever it may be. Whenever we are using the experimental animals, we need to take the permission from the IAEC, particularly for the small animals. And under the rules of breeding and experiment of animals, they are going to review our request. The request should be submitted in this form, that is Form B, we'll call it as, and it was notified by the CPCSEA. That means they have given this application. As per that, we have to submit the application, and IAEC is going to be give the permission to conduct the experiment under the guidelines. And another organization known as OECD, Organization of Economical Cooperation and Development. This OECD gives a guidelines for testing of chemicals. And these guidelines are unique tool for assessment or assessing the potential effect of chemicals on human health and environment. And these guidelines are accepted internationally as a standard method for safety testing. Guidelines are used by the professionals of industry, academia, as well as the government involved in the testing and assessment of chemicals. See, the OECD guidelines for testing the chemicals split into 5 different sections. In the beginning, the first one is the physical chemical properties. Second one is the effect of biotic system. Third one is environmental fate and behavior. Fourth one is the health effects. And the fifth one is the other test guidelines. See, we need to take the approval from the IAEC before we are going to conduct the experiment, and the experiments are conducted as per the CPCSEA guidelines. And we are going to select the models with the relevancy. That means based on whether we need the pharmacokinetic profile or pharmacodynamic responses. Then we need to select a specific species of the animal, sex and age group of the experimental group. And we are going to look into the susceptibility, sensitivity and reproducibility of the test system. And even in vitro also we are going to perform the toxicity test using isolated organs, tissue culture, etc. The toxicological studies are classified, that means in vivo toxicological studies. Under this, there are 3 different categories. The general test like short-term studies, acute, for example, acute toxicity tests to find out the lethal dose. Then long-term studies like subacute test, subchronic test and chronic test. And the second category of the toxicity study is the local toxicity test like dermatological preparations, and special toxicity tests like mutagenicity test, carcinogenicity test, reproductive and development test, then neurobehavioral toxicology and immunotoxicology, etc. See, these are the few different tests what we are going to conduct, like single dose, repeated dose toxicity. Yes. Single dose toxicity, repeated dose toxicity, local toxicity studies. Once again. Local toxicity studies, genotoxicity studies, then carcinogenicity or oncogenicity studies, then allergenicity or hypersensitivity toxicology studies. See, before going to the specific study, we should have some information about what are the clinical signs we need to observe during the study. Few are mentioned here, like respiratory, blockade in the nostril, that is change in the rate and depth of breathing, change in the color of the body surfaces, etc. We are going to observe. These are respiratory clinical signs. Then motor activity, changes in the frequency and nature of the movement, that like signs like decrease or increase in the spontaneous motor active movement, loss of the breathing reflex, anesthetic activity, catalepsy or unusual locomotion, tremor or fasciculation, like that. These are all CNS, sensory, autonomic and neuromuscular system working, affecting on the autonomic or neuromuscular system. Like that, some cases we are going to observe the convulsion, that is marked involuntary contraction or seizure of the contraction of the voluntary muscles we are going to observe, like clonic convulsion or tonic convulsion or tonic-clonic convulsion, respiratory failure, some cases neuromuscular and working on autonomic system. See, other signs like increase or decrease in the salivation, piloerection, analgesia, muscle tone, and in case of muscle tone, hypotonia or hypertonia, and GI signs like dropping faces, dropping fecus, emesis, and diuresis. See, let us see one particular test, that is single dose toxicity study. Here we are going to administer a single dose of the toxicant to an animal, and we are going to observe for 14 days if any changes in the morphological, biochemical or pathological conditions, or some cases the animal may going to die. In such cases we are going to dissect the animal and we are going to observe histopathological changes or histological changes in the dead animals. See, this is the protocol. The study period is around 14 days. Study on at least 2 different species we are going to conduct, like one rodent and one non-rodent, like mice or rat, or other category is the rabbit. A dose we are going to administer either orally or parenterally. A various dose level we are going to administer to the group of both the sexes, male and female. Dose selection in such a way that it should not be 0 death or it should not be 100% mortality. See, these acute oral toxicity guidelines, that is 401, in this study, the toxic characteristics of substance, acute oral toxicity testing is initial step. It gives the information about the health hazards. Test substances administered orally in a graduated doses to several group of the experimental animal. 1 dose used per group. At least 5 rodents at each group dose level of the same sex are used. Observations for effect and death are made. After the completion of the study in one sex, study in the other sex is carried out. Study suggested in rodent but can be adopted studies in the non-rodent. Here what we are going to do, step by step we are going to conduct this study. In the first, that means the toxic characteristic of the substance like oral toxicity.
We request everyone to kindly mute your microphone.
Test substances administered orally in a gradual doses to several groups. In the sense, for example, if we are taking 300 mg per kg for the 5 animals, then after completing that group, then we will move to the next group. Like that, there is a we are going to administer the doses for the different group, different doses. See, acute oral toxicity guidelines 420 is the other guideline. Here we are going to give the fixed dose. This particular study is done in 2 steps. One is sighting study. The second one is the main study. See here, you are going to understand. See, we are going to start this particular 420 guidelines by administering 300 mg per kg starting dose. The A indicates here the death of the animal, B is evident toxicity, and C is no evident toxicity and no death. See, for example, if you given 300 mg per kg to the animal, if the animal dies, then we are going to decrease the dose in the next group. Okay, then here also in the next group, if death of one animal occurs, then we are going to decrease the dose, that is to 5 mg. The beginning dose was 300 mg, then we are decreased to 50, then we are going to find the 5, that means 10 times we are decreased. Whereas if there is no death occurs, then we are going to increase to 200 mg per kg to one animal. In one animal, we are going to give the 200 mg per kg body weight. Then again, once if you administer 2000 mg per kg, if the death occurs, then we are going to decrease the dose. Same way, we are going to either decreasing or increasing will be done based on the condition of the animal. In the main study, here what we are going to do, we are going to add for the 5 animals 5 mg per kg. In the same manner, here we are going to A is again the 2 death, B is 1 with the evident toxicity and 1 with the death, or C is no evident toxicity. Here we are going to follow this chart to find out whether it is toxic or not, and again at what dose it is going to be toxic. Acute oral toxicity guidelines 1 to 3. Here no sighting study. 3 animals of single sex per step we are going to take. An average of 2 to 4 steps may be necessary to allow the judgment on the acute toxicity of the test substance. Not intended to allow the calculation of precise LD50. Here we are not going to calculate the LD50. The death of proportion of the animal as the major endpoint, and LD50 cutoff values are indicated here. The principle involved in this is stepwise procedure with the use of minimum number of animals per step, then substances administered orally to 2 groups of animals at a defined dose, and 3 animals per step of single sex, normally females we are going to select. Then compound related mortality determines the next step, and we are reporting at what dose it is showing the mortality. Acute oral toxicity guidelines number 4 to 5 is other guidelines. It is also known as up and down testing approach, was first described in Dixon and Moore. Here in 1985, use it for acute toxicity determination of chemicals, estimates the confidence interval of the LD50. Then in procedure, one animal dosed at the time at the minimum of 48-hour interval, suggested starting dose of 175 or can be selected from 1.75, 5.5, like that. There is an increase in the dose. Animal receive first dose a step below the level of the best estimated LD50, depending upon the outcome for the previous animal. The dose for the next animal is adjusted up and down. 5 reversal in 6 consecutive animals when obtained test is terminated. Number of animals limited to 15. Then we are going to conclude the dose. Here, there are several limitations for the toxicity study, indicated that all are likely to perform poorly for chemicals with the shallow dose response slope, because guidelines 420 uses evident toxicity as a endpoint instead of death. Usually, the test substances may cause delayed death, mostly in case of 4-5. However, both the guidelines 420 and 423, the finding of delayed death may require additional lower dose level to be used or study to be repeated.
You can go ahead, sir.
Okay. See, these are the short-term repeated dose toxicity studies like 407, 408, 410, 411, and 412 and 413. They have mentioned the numbers for the different studies and repeat. And here we are going to repeatedly administer the drug for a specified period of time. In this case, 2 mammalian species we are going to use a long duration. It is a long duration study, dose dependent on the dose escalating studies, then drug administered by clinical route. And what are the parameters we are going to measure here is the behavioral, physiological, and biochemical parameters. And finally, we are going to observe the histological characters, that is microscopic observations. Say like this, the different like 28 days, 90 days study, 1 rodent and 1 non-rodent species we are going to use, and the following parameters like weight variation, biochemical, cardiovascular parameters, behavioral changes, etc. we are going to observe. See here, once we administer the drug in the acute toxicity, if it is a 14-day study, we can able to find out the toxicity profile, that is LD50. In the subchronic study, that is a 90-day study, and no observable adverse events we are going to observe in this case, that is 1000 mg per kg. After performing that all the studies like biochemical parameters, histological study, etc. Then chronic study is a long-term study. The test compound is administered over more than 90 days and animals are observed periodically. The chronic toxicology study provides interference about the long-term effect of the test substance in the animals, and it may be extrapolated to the human safety of the test substance. The report on chronic oral toxicity is essential for new drug entities. There should be a little individual variation between the animals, and the allowable weight variation range is plus or minus 20 grams. See the types of the studies, that means it is a second category. Dermal toxicity studies, we are going to use rat and rabbits. Local signs like erythema, edema we are going to observe, and histological examination we are going to perform. Then dermal phototoxicity studies, for this we are going to use the guinea pig as the experimental animal used in the treatment of leukoderma, and here also we are going to observe erythema, edema formation. Then vaginal toxicity studies, the rabbit and the dogs are going to be used for this study. Observational like swelling and histopathology of vaginal wall they are going to perform. A rectal tolerance study, for this rabbit and the dog they are going to use, and the signs of pain, blood or mucus, then histology examination of rectal mucosa they are going to perform. Then ocular toxicity, albino rabbits we are going to use, or New Zealand white rabbit we are going to use. Changes in cornea, iris and aqueous humor they are going to estimate or observe. Then histological examination of the eye they are going to perform. Then for the parenteral drugs, for intravenous, intramuscular or subcutaneous or intradermal injection, site of injection examined grossly and microscopically. If any blebs or any wound occurs, then they will go for the microscopy. Then allergenicity or hypersensitivity toxicology study, guinea pig maximization test we are going to perform. determination of the maximum non-irritant or minimum irritant dose evaluation by erythema or the edema they are going to perform. Mice, local lymph node assay also done by mice of one sex they are going to use. Drug treatment given on ear skin, then auricular lymph node dissection done after the 5 days. Increase in 3H thymidine used for the evaluation.
You can go ahead.
Okay. The few in vivo tests we are going to perform, that is for chromosomal damage using the mammalian hematopoietic cell, then chromosome damage in the rodent hematopoietic cell using the micronuclear assay. Then carcinogenicity study are performed on drug used for more than 6 months or frequent intermittent use of chronic diseases. Chemical structure of the drug indicated, if the chemical structure of the drug indicated the carcinogenic potential, and therapeutic class of the drugs which have produced positive carcinogenicity, this class of the drugs will be tested for carcinogenicity study. A few in vitro toxicology study we are going to perform provides early indication of potential for some kind of toxic effect, allowing a decision to terminate the development of the program before spending the too much of money and the time. In vitro methods are widely used for screening and ranking of the chemicals, studying cell, tissue, or target specific effect, improve subsequent study design. Okay, thank you. Any questions?
Purti?
Thank you, sir. It was a very excellent session. Now, I would like to, before inviting the vote of thanks, I would like to read a few questions.
Yeah.
So there is this question which says, if you want to confirm drug against a disease in animal model, you should inoculate animal model with similar pathogen. The drug is not curable. What will you do next?
Pathogen we need to inject. That's what in the beginning I told you, we need to take permission from the animal ethical committee. Why I said, because they are going to screen, and finally what is going to happen, your question is, if the drug is not working in that particular pathogen or the inducer.
Yes, if the drug is not curable.
Not okay, not curable.
Yeah.
In such cases, after estimating, that means observing some parameters, it shows us if the disease is that means that particular conditions are decreasing or increasing, or if it is not affecting. In such cases, we need to sacrifice the animal. We need to sacrifice the animal. Otherwise, we are not going to do anything. But it should not harm to the other peoples.
Truly, yes.
Particularly, you are talking about microorganism. It comes under IBSC.
Okay.
It is not comes under IAEC.
Okay, sir. Thank you. Next question is, on what basis a chemical compound is considered to be toxic? Is there any threshold value for each clinical sign, or only LD50 value is considered?
LD50 itself an indicator. 50% of the animals are going to die at that particular dose, and we are going to find out the effective dose also, that is known as ED50. Once we know the ED50, that is for the therapeutic use, we are going to select the doses based on the ED50. LD50 is not a final value for us to treat. We are going to estimate the ED50 also.
Okay, sir. Next question is there any advances of avoiding animal-based studies and moving towards the system-based studies based on the current technology advancements in genetic engineering or IT or artificial intelligence, etc.?
Yes. In the one slide I have mentioned, the system is complex. We cannot create the system in such a complexity. Even we have the computers, as you mentioned, that is some biotechnology based, it is difficult for us to get all the complex condition. That means biochemical condition.
Yes, sir. Thank you. Last question is that what is the approximate success percentage of drugs entering to clinical studies from these animal model based preclinical studies?
Based on the preclinical, how many are moving to the clinical trials?
Yeah, exactly.
It is less only, because we know that before coming to the preclinical study, in vitro studies they are going to conduct. There only they are going to screen maximum through high-throughput screening and other things. Most of the cases, what is going to happen before coming to the preclinical itself, they are screened, and all the drugs what they are coming for preclinical may not move to the clinical. Percentage is very less only.
Okay, sir. Thank you so much. Now I would like to invite Vaishnavi Rao.
Spurti.
Sir.
Tantri is there. He wanted to ask one question, I think. He raised in the chat box.
Okay.
Tantri, you are there? Hello? Tantri? Yeah, maybe is mentioned. Okay.
Okay, sir. Now I would like to invite Vaishnavi Rao to deliver the vote of thanks.
Thank you, sir. Jay Kumar, sir.
Thank you, sir. Thank you, Ramesh. I would like to thank Dr. Ramesh, sir, Madam, and also the management of National College of Pharmacy. Thank you.
Thank you, sir.
Vaishnavi, are you there?
Yeah.
Yeah.
Good
Yes.
afternoon, everyone. I'm
Yeah.
honored and lucky to have the opportunity to give a vote of thanks on the special edition of international webinar conducted by our college. I'm very hearty gratitude to Dr. Vijay Kumar, sir, for sharing your graceful words on toxicological research on animal models. He has given us information about types of toxicity, its clinical signs, limitations of methods used for toxicity. Thank you, sir, for sparing time from your busy schedule and enriching us with your knowledge. Finally, I would like to thank our principal, speakers, teachers, organizing team, and all the participants for making this event a grand success. Thank you, one and all.
Thank you.
Thank you.
Thank you all once again. It gives a great pleasure and a privilege to say that we've got 1,200 registrants for this international webinar, which is, upholding for us at National College Baswangudi and also the National Education Society Karnataka. So on behalf of every one at our college, National College, we like to, thank everyone wholeheartedly for joining with us and making it a successful one. So we had, it's not a small job, to, bring on 7 speakers from different parts and with different idea and putting them into same topic of current trends and impacts and toxicants and biodiversity and human health. So this is such a great knowledge imbiber to all of us, and I wholeheartedly thank our zoology HOD Ramesh sir and also our principal of National College Dr. C.B. Annapurnamma and all the faculties for making it a very successful one, and also the participants who have registered with so much of interest, with so much of attainment to knowledge. So I would like to thank you all once again. I would now like to welcome Ramesh sir to say the vote of thanks.
No, madam, you have to invite Spurti. Spurti? Hello?
Principal madam.
Spurti? Spurti?
Hello.
Miss. She's got disconnected. Spurti is audible. Hello?
Sir, invite principal
Madam, good afternoon, madam. Good afternoon, madam. Kelsadya? Yes, ma'am. You can make a remark webinar, madam. Please. This is our college principal, Dr. C.B. Annapurnamma. So she is going to make that remark about this session or entire webinar.
Honorable Secretaries Professor SNR and Dr. P. Sadanandamaya, Professor P.L. Ramesh, HOD of Zoology, the organizer of this webinar, and his colleagues, respected resource persons, Dr. Prasad Gabita, Dr. Dinesh Kumar, Dr. Chandru Charvari Mat, Dr. Ravikansi, Dr. Viresh P. Virapur, Dr. Vijay Kumar, my dear students and colleagues and all the participants. Good afternoon to everybody. We are reaching the end of the success, no doubt. I congratulate all resource persons who delivered and thoughtful lectures on the respective topics. And thought-provoking for our students and also all the in his keynote address, he explained about Alzheimer's disease very well. From his lecture, we know there is improved medicine is coming for this disease. It is very happy thing. Dinesh Kumar explained about autophagy. He gave very good information about the respiratory mechanism and neuro inflammatory. This is very informative, sir. Chandru Charavarimat explained very special things. It is very astonishing to know how our respiratory system get affecting by farm culturing. Now we are thinking in different way. Nowadays, coliform, that curiform in our Karnataka, there is booming. Second way we are thinking, it is wise or we can continue this. Any precautionary measures we have to take for this. This is very informative lecture. And then P. Virapur, impacts of toxicants on human health. He explained about different toxicants effects like plastic, e-waste, and cosmetics also. He created very good awareness about this. Next, Dr. Ravikant, animal, plant, environment, and human health need for one program. ನನಗೆ ಈ ಸಂದರ್ಭದಲ್ಲಿ ದಾಸಿಮಯ್ಯನ ಒಂದು ವಚನ ಇಲ್ಲಿ ನನಗೆ ನೆನಪಿಗೆ ಬರ್ತಾ ಇದೆ. ನಾನು ಕನ್ನಡ ಲಿಟರೇಚರ್ ಸ್ಟೂಡೆಂಟ್, ಹಾಗಾಗಿ ನನಗಿದು ನೆನಪಿಗೆ ಬಂತು. ದಾಸಿಮಯ್ಯ ಈ ಜಗತ್ತನ್ನ ಕುರಿತು ಇದರ ಸೂತ್ರವನ್ನು ಕುರಿತು ಹೇಳ್ತಾನೆ, ಇದು ಜಗದ ಭಿನ್ನಾಣದೊಡಲು ಅಂತ ಹೇಳ್ತಾನೆ. ಈ ಪ್ರಾಣ ಪ್ರಕೃತಿಯೊಳಗೆ ನೀ ನಡಗಿಹ ಭೇದವನ್ನು ಲೋಕದ ಜಡಜೀವಿಗಳೆತ್ತಬಲ್ಲರೇ ರಾಮನಾಥ. ಜೈವಿಕ ಸೂತ್ರ ಏನಿದೆ, ಇದನ್ನ ಜಗದ ಭಿನ್ನಾಣದ ಒಡಲು, ಇದನ್ನ ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳೋದು ಬಹಳ ಕಷ್ಟ, ಇದರಲ್ಲಿ ಬಹಳ ವಿಸ್ಮಯ ಇದೆ ಅಂತ ಹೇಳ್ತಾನೆ. ಈಗ ಅನ್ಸುತ್ತೆ ಇವರು ಸರ್ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡಿದಾಗ ಇದನ್ನ ಕೇಳಿಸ್ಕೊಂಡಾಗ ಅನಿಸ್ತು, ಒಂದೊಂದು ಜೀವಿಯೂ ಸಹ ಈ ಜೈವಿಕ ಸೂತ್ರದಲ್ಲಿ ಒಂದೊಂದು ಕೊಂಡಿ ಇದ್ದಂತೆ. ಒಂದು ಕೊಂಡಿ ಕಳಚಿದ್ರು ಸಹ ಏನಾಗುತ್ತೆ, ಜೈವಿಕ ಸೂತ್ರ ಹರಿದು ಹೋಗುತ್ತೆ. ಇದನ್ನ ಅರ್ಥ ಮಾಡಿಕೊಳ್ಳುವಂತಹ, ಇದನ್ನ ಕಾಪಾಡಿಕೊಳ್ಳುವಂತಹ ಒಂದು ವಿಶೇಷವಾದ ತಿಳುವಳಿಕೆಯನ್ನ ಇವತ್ತು ವಿಜ್ಞಾನ ನಮಗೆ ದೊರಕಿಸಿಕೊಟ್ಟಿದೆ. ಇದನ್ನು ಕಾಪಾಡಿಕೊಳ್ಳುವ ಹೊಣೆಗಾರಿಕೆ ನಮ್ಮದಾಗಿದೆ. ಇದನ್ನ ಕುರಿತು ಶ್ರೀಯುತರು ತುಂಬಾ ಅತ್ಯುತ್ತಮವಾಗಿ ನಮ್ಗೆ ವಿಷಯವನ್ನ ತಿಳಿಸಿಕೊಟ್ಟಿದ್ದಾರೆ. ನೆಕ್ಸ್ಟ್ ವಿಜಯಕುಮಾರ್ ಅವರು ನಮ್ಮ ಪ್ರಯೋಗಶಾಲೆಯಲ್ಲಿ ಹೇಗೆ ಅನಿಮಲ್ ಮಾಡೆಲ್ಗಳನ್ನ ಬಳಸ್ತಾ ಇದೀವಿ ಅದನ್ನ ತಿಳಿಸ್ಕೊಟ್ರು. ಇದು ಬಹಳ ಟೆಕ್ನಿಕಲ್ ಸೆಷನ್. ನಮ್ಮ ಈ ಸಿಬಿಜೆಡ್ ವಿದ್ಯಾರ್ಥಿಗಳಿಗೆ ತುಂಬಾ ಉಪಯುಕ್ತವಾದಂತಹ ಮಾಹಿತಿ ಇದರಿಂದ ದೊರಕಿ. All these studies made this webinar great. I am appreciating their contribution. No doubt, the scientific research and innovations giving suitable solutions for our difficulties. It removes our anxiety, helps the qualitative life. For these reasons, the scientific invention should reach to everybody. In this context, appreciated that our zoology indulge students creative works in participating this webinar. I appreciate for their kind cooperation. In this lockdown period, my colleagues, Professor Ramesh and his team, Dr. Shakuntalama, and very good coordinator. I am and students. This is really, I am proud about you. Webinar.
Yes, Spurti?
Yes, sir. Last announcement, sir. The feedback forms are mentioned in the chat box. There are 3 simultaneous feedback forms which we have posted there. So if in case if the certificates do not reach you guys by today, all the registrants will get an email which contains these certificates, which contains the feedback form within 3 days time. And also these feedback forms will be posted in the WhatsApp groups. So simultaneously we will be taking on like 300 to 400 certificates per day. So please give us some days time, around like 22nd till 22nd. Please give us some time, and then if you do not receive the feedbacks till then kindly get back to us on mcbzoology@gmail.com. So those who have got your certificate, we wish you, we request you to support us for our future webinars, and also requesting everyone to again support like this in very much huge numbers. And then thank you all once again for making it a successful one. Yes, sir. Ramesh, sir, you're on mute.
You can invite the participant to speak a few words about this webinar.
Yeah, if anyone wants to share their thoughts on this webinar, you can please go ahead.
Shirodkar sir is there. Shirodkar sir. Yeah. Jairam sir.
Hello, Ramesh.
Good afternoon. Yes. Go ahead.
Sir, good afternoon, sir.
Good afternoon. Yeah.
Thank you very much for organizing such a wonderful webinar.
Thank you, sir.
We have very informative and a very nice presentation. All sessions are very good.
Thank you, sir.
Okay.
Shirodkar, sir. Yes, sir, Shirodkar. You wanted to judge it. Sir, where, sir, you want to spell few words?
Thank you very much, sir. Again, giving the opportunity to say something.
Yeah.
This webinar is of its own unique way. So it starts by morning 8 o'clock. I appreciate all your participants and your patience, all the organizing committee, your students, all sort of things. They've done very good arrangements, sir.
Yeah, thank you, sir.
Thank you very much.
Ravikant, yes. Ravikant, sir, can you?
Yeah, it was a very interesting conference.
So you only initiated, sir. You are the root causer for this webinar.
Thank you very much. I'm very happy to see so many people participating in it. Really, very happy. Yeah, thank you very much.
I think batchmates and is
Hello,
seen
Shirodkar, sir.
because of we got all these resource persons. Thank you, sir. Ravikant, sir.
Yeah, welcome.
Yeah, Shirodkar, sir. Yes.
Hello, sir. Hello, sir.
Yeah.
Really, it's a
Please.
great achievement, and thank you, Ramesh sir, to you
Yeah.
and your principal and your all the team workers, and one more thing, the resource persons are today very well they have presented and well informative and present what are the health problems are going on. No, at least we will come to know some the basic things regarding that. Really, it is wonderful and grand success, sir. Thank you, one and all, sir.
Thank you, sir. Yes, Madam Professor Swati Anjit, Madam, you want to speak?
Thank you very much.
Yes, Madam. You can unmute it. You can unmute and speak.
Yeah. It was a very nice, it was a pleasure to attend this webinar, sir. Very informative and very nice coordination, and really appreciate your effort and your students, how they have coordinated with this. Very nice. Thank you, sir.
Thank you, Madam. Thank you. If anybody want to speak, sir, other participants, faculty members are there, then.
Sir, if we can take a last group photo or a last 15-second video.
Yeah. Please.
I request you all to turn on your videos.
Videos, please. Gurudat and Navneet and others, please. Ravikant sir, you also can join.
Yeah, I've joined. Yes.
Yeah, Ravikant sir, you can also switch on your.
Yeah, switched on.
Yeah. Who are jointly there, the other two speakers. Yeah. And Nandini, madam, can you? Vaishnavi Rao, you also, you can. Vaishnavi Rao, yeah. You can switch on your, okay. Shabana, madam, please. Shabana Begum, madam. Shivshankar, you can also. Bhuvaneshwar, yes, Bhuvaneshwar. Okay. Yeah, Shivshankar and one more.
Can I start clicking, sir?
Yeah. Shivkumar, Dr. Shivkumar sir, can you want to speak few words? Yeah. Yes, sir. Go ahead, sir. Yes, you want to speak, sir? Unmute and speak, sir.
Sir.
Yeah.
Yeah. Thank you, sir. Very well organized. I'm very happy to be part of this international webinar, sir.
Yes, sir.
And all the speakers, they have spoken very well. And
Yes.
particularly your organizing team, I would like to thank. Wonderfully, your organizers.
Thank you, sir.
Thank you. Thank you very much, sir.
Yes. So we can, Spurti,
Yes, sir.
we can go to the vote of thanks, or.
Yes, sir. Okay. Now I would like to invite Ramesh sir to deliver the vote of thanks.
Yeah. Madam, matadbeka?
ಇಲ್ಲ ಕಟ್ ಆಯ್ತು ನಾನು ಮಾತಾಡ್ಬೇಕಾರೆ ಒನ್ಸ್ ಅಗೇನ್ ಐ ಆಮ್ ಥ್ಯಾಂಕಿಂಗ್ ಯು.
Okay, madam.
ಓಕೆ ಅದು ನಂದು GB ಅದು ಮುಗಿದೋಯ್ತು ಅದು.
Shakuntala madam, you ಹೇಳ್ಬೇಕಾ?
I thank all the participants for their patient listening and participated in the webinar so patiently. So, very good knowledge. This is for benefits. Thank you so much, sir. Thank you so much.
Okay. So
just a word from my side, if you don't mind, I think.
Yeah.
So
But
as we was attending from past 7:45 in the morning, so why we thought of as you instructed to write some keynote points and reports on that. As we have started noting for all those things, in our mind we just got one single note that we are not writing a report of a single webinar. We are writing the report of a PhD scholars or something like research reports. Barita Devi enhanced, sir. It was such an informative to such a range that we didn't expect, so that something will be there, but it was very good.
Really?
Sir, good afternoon, sir. Yashpal Singh, sir. You unmute, sir? You are from Jammu and Kashmir.
Good afternoon, sir. Good afternoon.
Good afternoon. Yeah, how are you, sir?
Sir, fine, sir. Very informative, sir. Babnon, sir.
Yeah. So you want to speak, sir?
Yes.
Yeah, you can speak, sir. Hello, Yashpal. Yashpal, sir. Go ahead, sir. Hello.
Seminar should be every year.
Voice is breaking. Sandeep, sir. Sandeep, sir, you can unmute and speak, sir. Yeah. Yeah.
Namaste, sir.
Namaste.
I'm very thankful to all of you who have arranged this webinar, wherein all the speakers were very informative, knowledgeable. I'm really thankful to all of you. Your teamwork is very fine. Thank you very much.
Thank you, sir. Okay. I think Shabana Begum is, Shabana Madam is there. Hello. Okay. Yes, Purti, you want to announce anything?
No, sir. I've mentioned everything about the feedback.
Sharda Sharma, Madam, can I know who you are from? Sharda Sharma.
Sir, good afternoon. I'm Sharda from Government Science College. Sir,
Ayo,
Bangalore.
sorry. That's what I am remembering your face.
Government Science College.
Yeah. No.
Yeah, I'm from Government.
Yeah.
Sir, it was very well organized. It's a team effort.
Yeah.
So all your students are like very good, sir. It was
Thank
a
you.
very good program. Thank you very much.
Thank you. Thank you, madam.
All the webinars are highly informative
Okay.
and research oriented. They have done it really well.
So,
I think you can give a chance to students, sir. Our
yeah,
students.
any students are there, they can speak few words. Student, Vaishnavi Rao or some who is there? Navneet is over. Sir, Narendra Kumar. Yeah, I think he is.
Yes, sir. Good afternoon, sir.
You are from
Jammu and Kashmir, sir.
Okay.
I am from Jammu and Kashmir. Okay, sir.
That's what, okay. Sorry. Yeah.
Yes, sir. The webinar which your team has conducted, it's very good, sir. Well organized, well planned, sir. Research oriented, really, sir, knowledgeable. Even your source persons are very good, sir. They have given us a very, you can say, information. Okay, mind-blowing webinars, sir. Eye-opening your webinars. Thank you, sir. Thank you very much. Thank you to your whole team, sir.
So,
sir, I request you further that you are.
Okay, so you both are from Jammu and Kashmir. Yeah, I remember. I think we met in the Meerut last time.
Okay, sir. Right, sir.
If I'm not wrong. Yeah. Okay, next.
Good afternoon.
So one from Uttarakhand was there.
Yeah, let her to speak. It's privilege. Now time is there anyhow. Yes. Where the Uttarakhand?
Hello, sir. Good afternoon.
Yes, madam.
So thank you for giving me an opportunity to participate in this webinar. I am a student of Maharani Lakshmi Ammani College for Women.
Yeah.
Really a very impressive and informative webinar. I am really thankful to you, Ramesh sir. I think you remember me. I had come to present a paper in your college last year.
2017.
Yes, sir. I won first prize in that.
Oh,
So that is when I met you, sir.
thank you. Thank you.
And hopefully, I think you remember Dr. R. Lakshmi Narayan.
Yeah, I remember.
I am his daughter who is speaking.
Oh, okay. See, pollution control board.
Yes, sir. Yes,
Yeah.
sir. Thank you very much for giving me an opportunity to take part in this webinar.
Thank you, madam.
And even your students, sir. Thank you very much.
Madhuri M, you want to speak, madam?
Yes, sir. It was a very informative session, sir. Lots of the resource persons were really extremely knowledgeable. It was nice for BSc also. It was very nice session, sir.
Thank you. Next. Advesh Kumar. Sir, you are from? Dr. Advesh Kumar. Sir, can you unmute and speak, sir? Advesh Kumar, sir. No, is. Sir, please unmute and speak, sir. Madam Kiran Mane Nagwada, madam, you are there? From Chikmagalur, I think. Okay. Okay, Spurti, we can go for the vote of thanks.
Yes, sir.
ಚೆನ್ನಾಗಾಯ್ತು ರಮೇಶ್. ಒಂದ್ ನಿಮಿಷ ತುಂಬಾ ಚೆನ್ನಾಗಿದೆ. ಕಂಗ್ರಾಚುಲೇಷನ್ಸ್ ಎಲ್ಲರಿಗೂ.
Okay. Thank you, madam.
ಥ್ಯಾಂಕ್ ಯು ಮೇಡಮ್. ನಮ್ಮ NAS ಪರವಾಗಿ ನಿಮಗೆ ಮತ್ತೊಮ್ಮೆ ಧನ್ಯವಾದಗಳು.
Advesh Kumar now is, sir, can you speak? No, ಗೊತ್ತಾಯಿಲ್ಲ. Okay. So it is a final step or last step of this webinar. It is my privilege to propose my vote of thanks. So it is my privilege to propose vote of thanks for everyone. First, I would like to extend my sincere thanks to Dr. H. Ramarao, President NAS of Karnataka, Professor S.N. Nagaraj Reddy, Dr. P. Sadanandamaya, Secretary NAS of Karnataka, for their encouragement and guidance. I thank Dr. C.B. Annapurnama, Principal, Dr. Y.C. Kamala, Secretary, College Council, National College, for support and help to organize this international webinar. I thank all the HODs, faculty, and students who are backbone of this event. I extend my warm thanks all the resource persons who are excelled in their respective research areas. I extend my sincere appreciation to each one of my associate and CBJ students who have worked hard to make this event a grand success. So my special or sincere thanks to all resource persons, especially from 3 resource persons abroad, United States of America, Dr. Prasant Gabita, Dr. Dinesh Kumar, Dr. Chandrasekhar Charyavarmat, Dr. Viresh P. Hirapur, Dr. Ravikant G. Bangalore, and Dr. Vijay Kumar. Yes, they are from Siddharth Pharmacy College. So I thank all the participants who are professors, institution heads, HOD, scientists, all the dignitaries, faculty from different disciplines from across Karnataka, across India, and even I think we have got approximately 25 to 30 USA registrations. So I thank all the abroad participants and participants from different states like Tamil Nadu, Kerala, Karnataka, Andhra, Rajasthan, UP, Jammu Kashmir, Kanyakumari to Jammu Kashmir. Kashmir to Kanyakumari. So we have all the participants. Thanks for attending this international webinar. I look forward to conduct this kind of webinars in coming days. And thanks for your attending from morning 7:45 to I think almost 2 p.m. now. I thank all the supporters, all the participants, and all the seniors, doctors, professors, institution heads. Thank you, one and all.
Okay, thank you, sir. Thank you.
Thank you, sir.
Thank you.
Thank you, sir.
Thank you, all of you.
Yeah. It is a great platform to communicate the faculty members or resource persons from one end to another end of the country or even other countries also. See, even the 3 US resource persons today, they only came to know that they are from India. So after attending this webinar or becoming resource persons for this, so I extend sincere thanks to each one of you, sir.
Principal madam. Hello. Paper ಕೊಟ್ರೆ writing ಇದು. Hello.
ಇಲ್ಲ ಮೇಡಮ್, ಇಲ್ಲ, ಅದು ಕೇಳಬೇಕು. We have to ask that thing.
ISBN ನಂಬರ್ ನಾವು ಅದನ್ನ ಪಬ್ಲಿಷ್ ಮಾಡ್ಸೋಣ. ನಂಬರ್ ರಿಸರ್ಚ್ ಸೆಂಟರ್ ಇಂದ.
Last ಮೇಡಮ್.
ಅದನ್ನೊಂದು ಮಾಡಿ.
ಆಯ್ತು. ಶಿರೋಡ್ಕರ್.
Sir, ನೀವು ಹೇಳ್ಬೇಕೀಗ. Actually, you have done a great job and wonderful.
I think within 15 days we have organized.
Yes. I know your effort and hard work along with your teamwork. Really,
short period of I think 7-8 days.
Yes. You have faced so many problems to get the link and all those things. But the resource persons are very well. They have presented very nice, sir. And it is within a short period. Really, you contact with all abroad peoples like the first 3 members of the webinar resource persons. Really, it is wonderful and excellent international webinar. Really, it is grand success, sir. Okay, all the best, sir.
Thank you, sir. Thank you.
And once again, I would like to remember Spurti, Navneet, and if you have such a students team, you can arrange one more in a grand success, sir. Okay.
Thank you, sir.
We have
Thank you so much, sir.
We have to extend our sincere thanks to our participants, sir.
Participants, yes.
Morning to till now, there are
huge number of participants. Really, it is a huge number. Really, it is wonderful, sir. Really.
Yeah, it is my privilege.
Yes.
Thank you.
Principal madam.
Hello.
Sir, Yashpal sir.
Principal madam.
Congratulations, madam. Second madam.
ನಿಮ್ಗೆ ಇಷ್ಟು ವಿಜ್ಞಾನದ ಬಗ್ಗೆ ಇಷ್ಟೊಂದು ಇಂಟ್ರೆಸ್ಟ್ ಇದೆ ನನ್ಗೆ ಗೊತ್ತಿದೆ.
Next time, Yashpal sir, then you unmute and speak.
ಅಲ್ಲ ಮೇಡಮ್, ನೀವು ಇಷ್ಟಪಟ್ಟು ಚೆನ್ನಾಗಿರೋದು
Jammu and Kashmir, sir.
Sir, allow to principal to talk, please. Allow to principal to talk.
Yeah.
ಮೇಡಮ್ ಆದ್ರೂ ಇಂಗ್ಲಿಷ್ ಬಗ್ಗೆ ಸೈನ್ಸ್ ಬಗ್ಗೆ ಎಷ್ಟೊಂದು ವಿಷಯ ತಿಳ್ಕೊಂಡಿದೀರಲ್ಲ ಮೇಡಮ್. ಆ ಇಂಟ್ರೆಸ್ಟ್ ಗೆ ನಾವು ಹ್ಯಾಟ್ಸ್ ಆಫ್ ಟು ಯು ಮೇಡಮ್.
ಅಲ್ಲ, ನೀವು ಇಷ್ಟು ಕಷ್ಟಪಟ್ಟು ಮಾಡ್ತಿದ್ದೀರಾ, ಇಷ್ಟು ಕಡಿಮೆ ಕಾಲಾವಧಿಯಲ್ಲಿ ಗ್ರಾಂಡ್ ಸಕ್ಸಸ್ ಮಾಡಿದೀರಾ ಅಂದ್ರೆ ನಾನು ಅದನ್ನ ಕೇಳಿಸ್ಕೊಳ್ದಿದ್ರೆ ಹೇಗೆ ಮೇಡಮ್ ಕುತ್ಕೊಂಡು?
ಅಲ್ಲ, ಕೇಳಿಸ್ಕೊಂಡಿದೀರಾ ಮೇಡಮ್, ಬಟ್ ಅದನ್ನ ಅರ್ಥ ಮಾಡ್ಕೊಂಡಿದೀರಲ್ಲ. ಕೇಳಿಸ್ಕೊಂಡು ಅರ್ಥ ಮಾಡ್ಕೊಂಡಿದೀರಲ್ಲ ಸೈನ್ಸ್ ಬಗ್ಗೆ.
ಸಿನ್ಸಿಯರ್ ನನ್ನ ಸಪೋರ್ಟ್ ಮಾಡ್ಬೇಕಲ್ವಾ ಮೇಡಮ್?
ನಿಮ್ ಕೋಆಪರೇಷನ್ ಮೇಡಮ್.
Sir, Yashpal sir, next time we are coming to Jammu and Kashmir.
Yes.
For conferences and seminars.
At your place, sir?
No, if Jammu Kashmir, not our place.
This time I am in Jammu, sir.
Yeah, I know.
Please come to Bangalore and visit our college. ಹೇಳಿ ರಮೇಶ್, ಕಾಲೇಜಿಗೆ ಬರ್ಲಿ, ಹೇಳಿ ಬೆಂಗಳೂರಿಗೆ ಬರ್ಲಿ, ಕಾಲೇಜಿಗೆ ಬರ್ಲಿ.
Pratibha madam, you want to speak? You are from madam?
Yashpal sir, they are inviting you to come to Karnataka, Bangalore.
Yes, Pratibha madam.
Redkar sir, you have invited me.
Definitely, sir.
Yes, sir.
Yes, sir. Pratima madam, can go
No,
ahead.
sir. No, sir. I'm very much thankful for
Okay.
you to allowing me for the late admission also. I didn't know the timing 8 o'clock.
Okay.
I joined very lately. Okay, sir. Thank you.
Okay, sir. Jairam sir.
Okay, thank you. Thank you very much, sir.
Thanks to all the participants once again.
Okay.
Thank you so much, sir.
College,
Ramesh, sir.
principal. Yeah.
Thank you so much, sir. Well organized and excellent
Yeah.
international webinar, sir.
Yeah. Thank you, madam.
Really, it was excellent.
I think Kiran madam is there. No, I think she left. Sushma Yadav.
Yes, sir. She was trying, actually. She was trying to talk to you.
Unmute ಮಾಡ್ಕೋಬೇಕಮ್ಮ.
Sir, thank you for organizing it. And I'm so happy that even in this lockdown, without colleges working, you have made our department to work together and made us to be successful in this.
I think 2017 national level conference, you are the...
She was a leader for that. She was a student leader.
Yeah, student leader.
Yes, sir.
Okay, thank you, Sushma.
It was a very good session, informative, and Chandra sir gave a very informative, and it is actually useful for this generation because we, this generation, are using sugar, salt, glutamate products more. So what they gave was very informative. Thank you.
Thank you so much, sir.
Okay, thank you.
Thank you, sir.
The platform to discuss all our knowledge and other ideas.
Yes, sir.
So I think.
Thank you, Sushma.
Thank you, ma'am. Thank you, sir.
I think more than 400 participants are frequently there in the webinar session. More than 400 participants from morning to till. Okay. So,
Okay, thank you. Thank you, sir. I'm leaving.
Yeah.
Thank you.
Yes, sir, we can end.
Okay. Okay, Ramesh.
Roshan Kumar is from, you wanted to, I think he is from Roshan. I think he is from UP. Yeah. Okay. Okay, Spurti. We can.
Okay, sir, we can leave. Thank you all once again. Keep supporting our national college.
One photo last one photo ತಗೊಂಡ್ಬಿಡೋಣ ಎಲ್ಲ ವಿಡಿಯೋ ಆನ್ ಮಾಡ್ಕೊಳ್ಳಕ್ಕೆ ಹೇಳ್ಬಿಡಿ.
Sir, ಆಯ್ತಲ್ವಾ ಸರ್?
Thank you, sir. Wonderful, sir.
ಯಾರು ಮಾತಾಡ್ತಾ ಇದ್ದಾರೆ? Okay. Thank you.
Okay, Ramesh.
Thank you, sir.
Thank you, sir. I am leaving now.
Yeah, thank you, sir.
I can leave, sir.
Yeah, sure.
Okay, sir.
Okay.
Sir, ಒಂದು 100 ಇಮೇಜಸ್ ಆಗಿದೆ ಫೋಟೋಸ್.
Okay, ಆಯ್ತು ಇರ್ಲಿ ಬಿಡಿ. ಇನ್ನೇನ್ ಸರ್ ಜಯರಾಮ ಸರ್
ಹೇಳ್ಬೇಕು.
ಇಲ್ಲ ಅಲ್ಲೇ ಇದ್ದೀರಾ?
ಚೆನ್ನಾಗಿತ್ತು. ಎಲ್ಲ ಚೆನ್ನಾಗಿತ್ತು. ಬೆಳಗ್ಗೆ 7:30 7:45 ಹಿಡಿದು ಈಗ 2 ಗಂಟೆ ಆಯ್ತು. ಆರ್ಗನೈಸಿಂಗ್ ಎ ನಂಬರ್ ಆಫ್ ಸೆಷನ್ಸ್ ಟುಡೇ. and all the sessions are very good, and you are organizing very nice in this international webinar. Thank you once again.
ನಮ್ಗೆ ಅದೇ ಪ್ರೌಡ್ ಈಗ, ಯಾಕಂದ್ರೆ US 25 ಜನ ರಿಜಿಸ್ಟ್ರೇಷನ್ ಆಗಿದ್ದಾರೆ. ನಾವ್ ಇನ್ನ ಮೊದಲು ಕಳಿಸ್ಲಿಲ್ಲ ಲಿಂಕ್. ನೆನ್ನೆ
ನೀವು ಲೇಟ್ ಆಗಿ ಕಳಿಸಿದ್ರಿ, ನಮಗೂ ಆದ್ರೂ ಲೇಟ್ ಆಗಿ ಕಳಿಸಿದ್ರಿ. ಬಟ್ ನೀವು ಸ್ವಲ್ಪ ಮೊದಲೇ ಕಳಿಸಿದ್ರೆ ಇಟ್ ಮೇ ಯೂಸ್ಫುಲ್ ಟು ಅದರ್ಸ್ ಆಲ್ಸೋ.
ಓಕೆ ಇರ್ಲಿ ಸರ್.
ಇರ್ಲಿ ಆದ್ರೆ ಇಷ್ಟು ಶಾರ್ಟ್ ಪೀರಿಯಡ್ ಅಲ್ಲೇ ಯು ಹ್ಯಾವ್ ಮೇಡ್ ಗ್ರ್ಯಾಂಡ್ ಸಕ್ಸಸ್ ದಿಸ್ ವೆಬಿನಾರ್.
ಓಕೆ ಸರ್,
ಓಕೆ ಸರ್.
ಓಕೆ, ಥ್ಯಾಂಕ್ ಯು.
ಓಕೆ, ಥ್ಯಾಂಕ್ ಯು. ಅದು ಇದು ಫೀಡ್ಬ್ಯಾಕ್
ಕೊಡ್ತಾರೆ, ಗ್ರೂಪ್ ಅಲ್ಲಿ ಹಾಕಿದ್ದಾರೆ. ನೀವು ಗ್ರೂಪ್ ಅಲ್ಲಿ ಇದ್ದೀರಾ? ಇಲ್ಲ,
ನೋಡ್ಬೇಕು.
ಗ್ರೂಪ್ ಅಲ್ಲಿ ಇದ್ರೆ ಇಲ್ಲ, ಎಲ್ಲರಿಗೂ ಸರ್ಟಿಫಿಕೇಟ್ ಬರುತ್ತೆ ಸರ್, ಡೋಂಟ್ ವರಿ.
ರಮೇಶ್ ಸರ್,
ಹೇಳಿ.
my special invitation to you. Next ಈಗ ಬರ್ಬೇಕಾದ್ರೆ with all your students you have to come over here. Ankola, yes, Ankola. Students.
You organize offline conference.
Yes, definitely. I'll try to organize offline. Then only you have to come here.
Yeah, sure.
All students who come, do come.
Yeah.
Okay.
Leave.
Okay.
Bye.
Okay, sir. Okay, bye. Bye, Shirodkar sir. Bye. Shirodkar sir.
Sir, ನೀವು ಬ್ಯಾಂಗ್ಳೂರಿಂದ ಅಟೆಂಡ್ ಆಗಿದ್ರಾ ಅಥವಾ ಧಾರವಾಡದಿಂದ ಅಟೆಂಡ್ ಆಗಿದ್ರೆ ಗೊತ್ತಾಗ್ತಲ್ಲ ನಮ್ಗೆ.
I am in between.
Okay, sir. Have a nice day.
I am in between Ankola and Bangalore. Okay, how are you? Shirodkar sir.
Fine, sir. Fine.
Avij, Aishu and Nikki.
Nice, sir. They are nice, sir.
ಮತ್ತೆ ಆರಾಮಲ್ಲ?
ಏನೋ ರಮೇಶ್ ಮಾತ್ರ ಒಂದು ಒಳ್ಳೆ ದೊಡ್ಡ ಜಾತ್ರೆ ಮಾಡ್ಬಿಟ್ಟಿದ್ರು ಪ್ಯಾಂಡಮಿಕ್ ಇದರಲ್ಲಿ.
ಹೌದು, ಅಲ್ಲಲ್ಲ, ಆಕ್ಚುಲಿ ಬಹಳ, ಯಾಕಂದ್ರೆ ಬೆಳಿಗ್ಗೆಯಿಂದನೂ ಇಲ್ಲಿವರೆಗೂ ಮತ್ತೆ ಎಲ್ಲಾ ಸೆಷನ್ಸ್ ಒಂದಕ್ಕಿಂತ ಒಂದ್ ಚೆನ್ನಾಗಿದ್ವು.
ಅಂತಾರಾಷ್ಟ್ರೀಯ ಜಾತ್ರೆ,
ರೆಲೆವೆಂಟ್
ಕಾಲೇಜಿನ ಬಸವನಗುಡಿ ನ್ಯಾಷನಲ್ ಕಾಲೇಜಿನ ಅಂತಾರಾಷ್ಟ್ರೀಯ ಜಾತ್ರೆ, ಇದು ದೊಡ್ಡ ಜಾತ್ರೆ ಈ ಪ್ಯಾಂಡಮಿಕ್ ಇದರಲ್ಲಿ. ರಿಯಲಿ ಗ್ರೇಟ್ಫುಲ್ ಟು ಯು ಸರ್.
ನಿಮ್ಮದು ಇದೊಂದು ಮಿಸ್ ಆಯ್ತು, ಅಸೋಸಿಯೇಷನ್ ಒಂದು ಮಿಸ್ ಆಯ್ತು.
ತೊಂದ್ರೆ ಇಲ್ಲ ಸರ್,
ನೆಕ್ಸ್ಟ್ ಟೈಮ್ ಮಾಡೋಣ.
ಓಕೆ ಸರ್.
ಸರ್ಟಿಫಿಕೇಟ್ಸ್ ಯಾವ ರೀತಿ ಯೂಸ್ ಆಗುತ್ತೆ ಅಂತ ಹೇಳಿ ಸರ್.
ಯಾರು ಮೇಡಮ್? ಇದು ದಿ ಸರ್ಟಿಫಿಕೇಟ್ ಈಸ್ ಆಸ್ ಸಿಮಿಲರ್ ಟು ದಿ ಸರ್ಟಿಫಿಕೇಟ್ issued by the offline. Because during this COVID pandemic, you can't conduct the offline sessions programs. So definitely they agree or accept this kind of certificates. No doubt at all. So actually we plan to call a research papers or oral presentation or poster presentation, but we didn't do that because lack of time and other aspects. So I think in our next webinar we'll do that.
Thank you, sir.
Okay. So chat box program. Okay, Shakuntala madam.
Yes, sir. Thank you, sir. Yes, sir. Ego.
Avani namma nala Dr. Sandeep Kaur. May I know Sandeep Kaur? You're there. Dr. Sandeep Kaur. Can you speak? May I know? Dashrit Kaur, you are there? Dashrit Kaur. I think she is from Punjab. Dashrit Kaur, please unmute and speak. She is from Punjab. I know. I think Punjab or Haryana. Okay. No, I think. Okay. Bye. Thank you.
Bye, sir. Bye.
Bye. Bye to everyone.